in the beginning, there was nothing. Then, somehow, impossibly, there was everything. But between that cosmic birth and the moment you opened your eyes this morning lies one of the most profound mysteries science has ever encountered. How did lifeless matter become alive? Picture Earth 4 billion years ago, a hellish world of molten rock, poisonous atmosphere, and relentless bombardment from space. No trees, no grass, no birds singing at dawn, just chemistry, raw, violent, beautiful chemistry. Yet from this chaos emerged something extraordinary, something that could grow, reproduce, evolve, and eventually contemplate its own existence. Something that would one day build telescopes to peer into the cosmos and ask the ultimate question: are we alone? This is the mystery of abiogenesis, the origin of life from non-living matter. It stands as one of science's greatest unsolved puzzles, right alongside the nature of consciousness and the fate of the universe itself. And like all great mysteries, it has spawned theories both elegant and wild, experiments both brilliant and controversial, and debates that rage in laboratories and lecture halls around the world. For centuries, humans believed life simply appeared spontaneously. Meat left in the sun bred maggots. Mud created mice. Life seemed to emerge from death as naturally as flowers bloom in spring. But science has a way of shattering comfortable assumptions. In the 17th century, Francesco Redi showed that maggots came from flies, not from rotting meat. In the 19th century, Louis Pasteur demonstrated that even microscopic life required parents. Spontaneous generation, it seemed, was dead. But this created a paradox. If life cannot arise from non-life, how did it arise in the first place? The answer had to lie in the deep past, when Earth was young and the rules were different. Scientists began to imagine a time when chemistry might have crossed the threshold into biology through processes we could barely imagine. The first serious attempt to recreate this ancient alchemy came in 1952 in a laboratory at the University of Chicago. Stanley Miller, then a graduate student working under Harold Urey, constructed what would become one of the most famous experiments in the history of science. He built a miniature primordial Earth in glass flasks and tubes, filling them with the gases he believed composed the early atmosphere: methane, ammonia, hydrogen, and water vapor. No oxygen, for oxygen would have been a poison to early life, just as it nearly destroyed all life when photosynthetic bacteria first began producing it billions of years ago. Miller then did what nature might have done. He sparked his mixture with electrical discharges, mimicking the lightning that would have split the ancient skies. For a week, his apparatus bubbled and sparked, creating a symphony of chemical reactions. When he finally analyzed the results, Miller made a discovery that sent shockwaves through the scientific community. His simple mixture had produced amino acids, the building blocks of proteins, the molecular machines that drive all life. The Miller-Urey experiment was revolutionary because it showed that the molecules of life could arise naturally from simple ingredients and energy sources that existed on the early Earth. In the decades that followed, scientists expanded on Miller's work, creating increasingly complex organic molecules in laboratory conditions designed to mimic the primordial world. They produced nucleotides, the building blocks of DNA and RNA. They created fatty acids that could form primitive cell membranes. They even synthesized molecules that could catalyze their own formation, creating the first hints of self-replicating chemistry. These experiments painted a compelling picture of life's emergence. Perhaps it began in tidal pools warmed by the sun, where organic molecules could concentrate and interact. Perhaps it started near hydrothermal vents on the ocean floor, where mineral-rich water provided both energy and a protective environment. Perhaps it arose in clay deposits, where minerals could serve as templates for organizing complex molecules. The possibilities seemed endless, and each new discovery added another piece to the puzzle. Yet for all their elegance and insight, these experiments revealed a troubling truth. Creating the building blocks of life is one thing, creating life itself is quite another. The gap between a soup of organic molecules and even the simplest living cell remains vast, perhaps unbridgeably so. Even the most primitive bacteria are fantastically complex, with thousands of different proteins working in perfect harmony, DNA storing and transmitting information with digital precision, and metabolic networks more sophisticated than any chemical plant humans have ever built. This is known as the complexity problem, and it haunts every theory of abiogenesis. How do you get from a random mixture of molecules to the intricate, self-sustaining, self-replicating systems we call life? It's like expecting a tornado in a junkyard to assemble a working airplane, except the airplane must also be able to reproduce itself and evolve into better airplanes over time. Some scientists have proposed solutions. Maybe life began with simpler self-replicating molecules, like certain types of RNA, that gradually evolved greater complexity. Maybe it started with metabolic networks that could maintain themselves before developing the ability to reproduce. Maybe it required specific mineral surfaces that could organize and concentrate organic molecules in just the right way. Each theory has its proponents and its problems, but none has achieved the kind of breakthrough that would solve the puzzle once and for all. The challenges go beyond mere complexity. There's also the problem of time. Life appears in the geological record remarkably early, within the first few 100 million years after the Earth became habitable. That sounds like a long time, but in chemical terms, it's almost impossibly brief. The Earth had barely cooled from its fiery birth when life was already leaving its mark in ancient rocks. This rapid appearance suggests either that life arises more easily than we think, or that something else was at work. Then there's the problem of conditions. The early Earth was not the gentle, nurturing environment that popular imagination often depicts. It was a world under assault, bombarded by asteroids and comets in what scientists call the Late Heavy Bombardment. The atmosphere was poisonous, the oceans were acidic, and the sun was 30% dimmer than it is today. Yet somehow, in this hostile environment, the most unlikely event in cosmic history supposedly occurred. Life began. These challenges have led some scientists to question whether Earth alone could have been the birthplace of life. Maybe the chemistry was too complex, the conditions too harsh, the time too short. Maybe something was missing from the Earth-only picture, some crucial piece of the puzzle that we haven't considered. Maybe life didn't begin here at all. This is where our story takes an unexpected turn, from the familiar realm of terrestrial chemistry to the vast expanses of cosmic space. What if the origin of life is not an Earth-only story? What if it's a tale that spans solar systems, involves entire galaxies, and connects us to the deepest mysteries of the cosmos? What if life itself is a cosmic phenomenon, seeded throughout the universe by processes we're only beginning to understand? Enter panspermia, one of the most controversial and captivating ideas in all of science. It suggests that life on Earth didn't arise here from scratch, but instead arrived from somewhere else, carried across the void by natural processes we're still discovering. It's an idea that challenges our most basic assumptions about our place in the universe and forces us to confront the possibility that we are not alone and never have been. Panspermia doesn't solve the ultimate question of how life began. It simply moves the problem to another time and place. But in doing so, it opens up new possibilities, new ways of thinking about life's origin that might finally provide the breakthrough we've been seeking. It suggests that the universe itself might be alive in ways we never imagined. That life is not a rare accident, but a fundamental feature of cosmic evolution, as natural and inevitable as the formation of stars and galaxies. This idea transforms everything. If panspermia is correct, then Earth is not a unique oasis in a sterile cosmos, but one of countless worlds touched by the same living processes. We are not alone, struggling to understand our origins in isolation, but part of a vast community of life that spans the universe. Our ancestors are not just the first cells that arose in some primordial pond, but the cosmic processes that brought life itself into being among the stars. The implications are staggering. Every meteor shower becomes a potential delivery of new genetic material. Every comet carries the seeds of worlds yet to be born. Every planet orbiting a distant star might harbor life that shares our deepest ancestry. We are not just children of the Earth, but children of the cosmos itself, connected to every living thing that has ever existed or ever will exist throughout the universe. But extraordinary claims require extraordinary evidence, and the evidence for panspermia remains tantalizingly incomplete. Like a detective story with crucial clues still missing, the case for life's cosmic origin rests on circumstantial evidence, suggestive hints, and tantalizing possibilities rather than definitive proof. Yet the story it tells is so compelling and the implications so profound that it has captured the imagination of scientists and the public alike. In the chapters that follow, we'll explore this cosmic mystery in all its complexity. We'll journey through the history of the panspermia hypothesis, from its ancient origins to its modern scientific formulations. We'll examine the harsh realities of space and ask whether life could possibly survive the journey between worlds. We'll investigate the evidence from meteorites and comets, the tantalizing hints of life's cosmic connections that have been falling from the sky for billions of years. We'll visit Mars in its ancient glory when it possessed oceans and an atmosphere, and ask whether the red planet might have been life's first home in our solar system. We'll explore the possibility that advanced civilizations might deliberately seed life throughout the cosmos, and examine what this might mean for our own future and our responsibilities to the universe. We'll confront the limits of what science can tell us and acknowledge the vast unknowns that still surround life's deepest mysteries. Most importantly, we'll grapple with what panspermia means for our understanding of ourselves and our place in the cosmos. Whether life began on Earth or arrived from elsewhere, the implications reshape our entire worldview. If we are alone, we bear a tremendous responsibility as the universe's only known conscious observers. If we are part of a vast community of life, we are connected to something far greater than ourselves, participants in a cosmic story that began with the first stars and will continue until the last one dies. This is not just a scientific question, but a philosophical and even spiritual one. It touches the deepest parts of human nature, our need to understand where we come from and why we're here. It speaks to our loneliness and our hope, our fear of isolation, and our dream of connection. It challenges us to think beyond our planetary boundaries and imagine ourselves as truly cosmic beings, shaped by processes that dwarf our everyday experience, but that nevertheless flow through our very atoms. The journey we're about to embark upon will take us to the edge of scientific knowledge and beyond, into realms where speculation meets rigorous investigation, where ancient wisdom encounters cutting-edge research, where the profound and the practical intersect in ways that might reshape our understanding of life itself. It's a journey that begins with a simple question: how did life begin, but ends with insights that could transform our species and our relationship with the cosmos? Are you ready to explore the possibility that we are all aliens, descendants of life that arose not on Earth, but among the stars themselves? Are you prepared to consider that every breath you take contains atoms forged in stellar furnaces and perhaps once part of living beings on worlds beyond imagination? Can you imagine a universe where life is not rare but common, not isolated but connected, not accidental but inevitable? If so, then let's begin this cosmic detective story. this exploration of one of the most profound questions ever asked. Let's discover whether life on Earth is a unique miracle or part of something far grander, a cosmic web of living processes that connects every world, every star, every galaxy in an unbroken chain of existence that stretches back to the very beginning of time itself. The word panspermia comes from ancient Greek, meaning seeds everywhere. It suggests that life spreads throughout the universe like dandelion seeds on a cosmic wind, taking root wherever conditions allow. This isn't modern science fiction. It's an idea that has haunted human imagination for over 2,000 years. In 5th century Greece, while most philosophers debated the nature of earth, air, fire, and water, a radical thinker named Anaxagoras proposed something far stranger. He suggested that the seeds of all things, including life itself, existed everywhere in the cosmos. These seeds, he claimed, could combine in infinite ways to create the diversity we see around us. Anaxagoras was eventually exiled from Athens for his revolutionary ideas, but his concept of cosmic seeds would echo through centuries of human thought. The Greeks weren't alone in imagining life's cosmic connections. Ancient Hindu texts speak of life existing throughout the universe in countless forms. Islamic scholars of the medieval period wrote about the possibility of life on other worlds. But it would take until the modern era for these philosophical speculations to transform into scientific hypotheses that could be tested and examined. The real birth of scientific panspermia came in 1865, when German physicist Gustav Richter made a startling proposal. Meteorites, he suggested, might carry living organisms between planets. This wasn't just poetic speculation, it was a testable scientific claim. Richter had noticed that meteors seemed to come from the cold depths of space, where extreme temperatures might preserve life in a kind of suspended animation. When these frozen travelers crashed into warm, wet planets, life might awaken and begin to flourish. But it was Swedish chemist Svante Arrhenius who truly launched panspermia into scientific respectability. In 1903, Arrhenius calculated something remarkable. He showed that tiny spores, pushed by radiation pressure from stars, could actually travel between solar systems. Light itself, he realized, exerts a gentle but persistent pressure on small particles. Given enough time, this stellar wind could carry microscopic life across the galaxy. Arrhenius called his theory radiopanspermia, and he worked out the physics with characteristic precision. A spore just the right size, he calculated, could be accelerated to tremendous speeds by starlight. It could escape the gravity of its home planet, survive the journey through space, and eventually be captured by another world. The voyage might take hundreds of thousands of years, but for a dormant spore, time would be irrelevant. The Swedish scientist envisioned vast clouds of living particles drifting between the stars, carried by currents of light and gravity we were only beginning to understand. Some would be destroyed by cosmic radiation or extreme temperatures. Others would drift forever in the emptiness between galaxies. But a few, a precious few, would find new homes on distant worlds, bringing with them the spark of life. Arrhenius faced fierce criticism from his contemporaries. How could delicate living organisms survive the brutal environment of space? How could they withstand radiation levels that would kill any known life form? How could they navigate the vast distances between stars without being destroyed or lost forever? These questions haunted early panspermia research and continue to challenge scientists today. The idea might have died there, dismissed as an interesting but impossible theory, if not for two British scientists who would revive it with new evidence and modern insights. In the 1970s, astronomer Fred Hoyle and mathematician Chandra Wickramasinghe began studying interstellar dust through powerful telescopes. What they found astonished them. The dust wasn't just rock and ice, as most astronomers assumed. Spectroscopic analysis revealed complex organic molecules, the chemical building blocks of life itself. Even more surprising, some of these molecules seemed to absorb and emit light in patterns that resembled biological materials. Hoyle and Wickramasinghe began to suspect that space wasn't sterile at all, but teeming with organic chemistry. They took their hypothesis further than anyone before them. Not only could life travel between worlds, they argued, but it might actually originate in space itself. They proposed that life first arose in the dense molecular clouds that drift between stars, where organic chemistry could proceed for millions of years without interference. These cosmic nurseries, they suggested, were the true birthplace of life in the universe. Hoyle and Wickramasinghe pointed to astronomical observations that seemed to support their radical theory. They noted that the infrared signatures of interstellar dust matched remarkably well with dried bacteria and other biological materials. They argued that comets, which formed from this cosmic dust, should be rich in organic compounds and possibly even living organisms. They even suggested that some epidemic diseases on Earth might be caused by viruses arriving from space. Their ideas were revolutionary and controversial. Most scientists remained skeptical of such extreme claims, but Hoyle and Wickramasinghe had done something crucial. They had updated panspermia for the space age, providing new evidence and new mechanisms that made the theory scientifically testable. As space exploration advanced, scientists began to recognize that panspermia might take many different forms. Radiopanspermia, Arrhenius's original concept, remained one possibility, but there were others, each with its own advantages and challenges. Lithopanspermia emerged as perhaps the most plausible variant. This theory suggested that life could travel inside rocks, protected from the harsh environment of space by layers of stone and metal. When asteroids or comets collided with planets, they could blast material into space at tremendous speeds. If any of this ejected material contained living organisms, they might survive the journey and eventually land on other worlds. The physics of lithopanspermia were compelling. Powerful impacts could indeed launch rocks into space at escape velocity. These natural spacecraft could travel for millions of years through the solar system and beyond. When they finally encountered another planet, they could deliver their biological cargo intact, protected by their rocky shells from radiation and temperature extremes. Computer simulations showed that such transfers were not only possible, but inevitable. Every major impact in solar system history would have exchanged material between planets. Mars and Earth, in particular, had almost certainly traded rocks back and forth for billions of years. If life existed on either world, it could have spread to the other through this natural exchange program. Scientists also began to consider more exotic forms of panspermia. Perhaps life could survive inside comets, frozen in ice for millions of years until solar heat awakened it. Perhaps it could drift as microscopic particles in the solar wind, carried by magnetic fields and gravitational currents. Perhaps it could even hitch rides on spacecraft, spreading inadvertently as humans began to explore the cosmos. The most controversial variant was directed panspermia, first proposed seriously by Francis Crick, co-discoverer of the structure of DNA. Crick suggested that advanced alien civilizations might deliberately seed life throughout the galaxy, sending specially designed organisms to promising planets. This wasn't science fiction speculation, but a serious scientific hypothesis from one of biology's most respected figures. Crick argued that directed panspermia could explain some puzzling features of life on Earth. Why did all terrestrial life use the same genetic code? Why did it prefer certain chemical forms over others that seemed equally viable? Perhaps because it all descended from organisms designed by a single advanced civilization, carrying the biochemical signatures of their alien creators. The directed panspermia hypothesis opened up fascinating possibilities. Maybe Earth was part of a vast biological project, seeded by beings we couldn't even imagine. Maybe our entire evolutionary history was guided by alien intelligence. Maybe we ourselves were the products of cosmic engineering on a scale that dwarfed human comprehension. But directed panspermia also raised disturbing questions. If aliens had seeded Earth, where were they now? Had they died out, leaving us as their only legacy? Were they still watching, waiting for us to mature enough for contact? Or were we just one experiment among millions, too insignificant to merit their attention? Each variant of panspermia offered its own insights and challenges. Radiopanspermia provided a natural mechanism for spreading life, but required organisms to survive naked exposure to space. Lithopanspermia offered protection, but limited the size and type of organisms that could travel. Directed panspermia could explain life's universal features, but required the existence of advanced alien civilizations. Modern panspermia researchers have embraced this diversity, recognizing that life might spread through multiple mechanisms operating simultaneously. Perhaps tiny spores ride stellar winds between nearby systems, while larger organisms travel in meteorites within solar systems. Perhaps natural processes dominate in some regions of the galaxy, while intelligent seeding occurs in others. Perhaps the universe employs every possible method to spread life as widely as possible. This multiplicity of mechanisms makes panspermia both more plausible and more difficult to test. If life can travel by many different routes, then evidence for any single route might be subtle and indirect. Researchers must look for patterns and signatures that emerge from the combined effects of all possible transfer mechanisms. The modern panspermia hypothesis also embraces a temporal dimension that earlier versions lacked. Life might have been spreading throughout the galaxy for billions of years, creating vast networks of related organisms across countless worlds. Each planetary biosphere might contribute its own innovations to this galactic gene pool, while also receiving new genetic material from other worlds. This vision transforms panspermia from a simple transfer mechanism into a theory of cosmic evolution. Life becomes not just a planetary phenomenon, but a galactic one, with individual worlds serving as nodes in a vast biological network that spans space and time. Evolution operates not just on single planets, but across entire star systems, with genetic information flowing between worlds like rivers joining an ocean. The implications are profound. If panspermia operates on galactic scales, then life on Earth might be connected to organisms thriving around distant stars. Our evolutionary history might include chapters written on worlds we've never seen. Our future might depend on biological innovations arising in planetary laboratories light years away. This cosmic perspective on life also suggests new approaches to searching for extraterrestrial organisms. Instead of looking for completely alien life forms, we might find organisms that share fundamental biochemical features with terrestrial life. We might discover that life throughout the galaxy uses similar genetic codes, similar metabolic pathways, similar cellular structures. The universe might be far more biologically unified than we ever imagined. But panspermia also raises the ultimate question: where did life begin in the first place? Moving the origin from Earth to space or to other worlds doesn't solve the fundamental mystery of abiogenesis. Somewhere, somehow, non-living matter became alive for the first time. Panspermia suggests that this momentous event might have occurred not on Earth, but elsewhere in the cosmos, perhaps under conditions we haven't yet imagined. The search for life's ultimate origin might require us to think beyond individual planets or even individual solar systems. Perhaps life arose in the dense molecular clouds that form stellar nurseries. Perhaps it began in the plasma surrounding young stars. Perhaps it emerged from quantum processes in the vacuum of space itself. The possibilities seem as endless as the cosmos that contains them. As we prepare to examine whether life could actually survive the journey between worlds, we must remember that panspermia is not just about transportation mechanisms or survival strategies. It's about reimagining life itself as a cosmic phenomenon, as fundamental to the universe as gravity or electromagnetism. It suggests that we live not on an isolated world, but as part of a vast living system that encompasses the entire galaxy and beyond. Whether this cosmic vision proves correct remains to be seen. But the very possibility has already begun to transform how we think about life, evolution, and our place in the universe. We are no longer just children of Earth, but potential citizens of the cosmos, connected to every living thing that has ever existed or ever will exist among the stars. Imagine stepping outside without a spacesuit 400 kilometers above Earth. The International Space Station hurtles beneath you at 27,000 kilometers per hour, but you barely notice because you're already dying. Your skin doesn't explode like in the movies. Instead, something far stranger happens. The saliva on your tongue begins to bubble and foam. Your blood doesn't boil, but the dissolved gases in your tissues form tiny, agonizing bubbles. You have perhaps 15 seconds of useful consciousness before the vacuum claims you. This is what space does to complex life forms. But what about a single bacterial cell, smaller than the width of a human hair? What happens when the cosmos strips away every protection that Earth provides and exposes microscopic life to the raw fury of the universe? The answer isn't what most people expect. A bacterium experiences vacuum differently than a human. With no lungs to collapse and no blood to outgas, pressure loss becomes almost irrelevant. The cell membrane might stretch slightly, but the internal pressure in most bacteria is surprisingly low. Some species spend their entire existence in low-pressure environments and show no distress when pressure drops to zero. The vacuum that kills us in minutes might barely register to them. Temperature presents a more complex challenge. Between the stars, space reaches temperatures so cold that atoms barely vibrate. 2.7 degrees above absolute zero. At such temperatures, chemical reactions essentially stop. For most organisms, this means death. But for others, it means something else entirely: perfect preservation. In 1995, Russian scientists extracted bacteria from permafrost that had been frozen for 3 million years. When thawed and provided with nutrients, these ancient microbes began growing and reproducing as if they had been dormant for mere days. The extreme cold hadn't killed them. It had simply pressed the pause button on their biological processes. Space cold operates the same way, but more effectively. The near absolute zero between stars could preserve organisms for billions of years without significant deterioration. Time itself becomes meaningless to a frozen spore drifting in the cosmic deep. It could traverse entire galaxies while remaining perfectly viable, waiting for the warmth of a new star to awaken it. But space doesn't offer uniform cold. Solar radiation can heat objects to hundreds of degrees Celsius. A meteorite passing close to a star might experience surface temperatures that would melt copper. Yet even this presents survivable challenges for the right organisms. In 1977, scientists discovered bacteria living in the water cooling systems of nuclear reactors, thriving in temperatures exceeding 80 degrees Celsius. Other species survive in deep-sea volcanic vents where water reaches temperatures of over 400 degrees Celsius, prevented from boiling only by enormous pressure. These extremophiles prove that life can adapt to temperature ranges once thought impossible. The real killer in space isn't temperature or vacuum, it's radiation. Every second, cosmic rays sleek through the solar system at nearly the speed of light. These aren't rays in the traditional sense, but atomic nuclei accelerated to impossible velocities by magnetic fields around distant stars. When a cosmic ray strikes a living cell, It tears through DNA like a microscopic bullet, leaving a trail of molecular destruction in its wake. The numbers are staggering. In deep space, radiation levels exceed Earth's background radiation by factors of hundreds or thousands. A human exposed to such levels would develop fatal radiation sickness in hours. Genetic material would fragment, proteins would denature, cell membranes would lose their ability to maintain the delicate chemical gradients essential for life. But some organisms laugh at radiation levels that would sterilize medical equipment. Deinococcus radiodurans earned the nickname the toughest bacterium on Earth after surviving radiation doses 5,000 times higher than what kills humans. Its secret lies in redundant copies of its genetic material and incredibly efficient DNA repair systems. When radiation tears its genome apart, the bacterium simply rebuilds itself from the scattered pieces, often within hours. Even more remarkable is Thermococcus gamma-tolerans, discovered in deep-sea sediments. This organism continues to grow and reproduce in radiation fields so intense they would destroy most laboratory instruments used to measure them. Its cellular repair mechanisms work so efficiently that radiation damage never accumulates faster than it can be fixed. These discoveries suggest that radiation, while dangerous, might not be the absolute barrier to space travel that scientists once believed. The question becomes whether such hardy organisms could maintain their repair systems during the thousands or millions of years required for interplanetary or interstellar journeys. This is where space agencies began conducting the most ambitious biology experiments in human history. The European Space Agency's EXPOSE platform has exposed hundreds of biological samples to raw space conditions outside the International Space Station. Unlike the protected environment inside the station, these experiments face direct vacuum, temperature cycling from -150 to +120 degrees Celsius, and radiation levels thousands of times higher than Earth's surface. The results have been both sobering and encouraging. Bacillus subtilis spores, among the most radiation-resistant organisms known, survived 18 months of space exposure with minimal shielding. When returned to Earth and provided with nutrients, many began growing normally. Their DNA showed damage, but their repair systems had prevented complete genetic collapse. Lichens, complex partnerships between fungi and algae, survived even longer exposures. After spending a year and a half in space, many samples resumed photosynthesis within days of returning to favorable conditions. The organisms had essentially hibernated through conditions that would kill most Earth life within minutes. But the real celebrities of space survival research are tardigrades, microscopic animals that look like eight-legged bears under a microscope. These creatures possess an almost supernatural ability to survive conditions that destroy other life forms. When faced with extreme stress, tardigrades enter cryptobiosis, a state so profound it barely qualifies as life. They expel nearly all water from their bodies, reducing their metabolism to less than 0.01% of normal levels. In this state, they can survive temperatures from near absolute zero to over 150 degrees Celsius, radiation levels exceeding those found near nuclear reactors, and pressures 6 times greater than those in Earth's deepest ocean trenches. In 2007, the European Space Agency launched thousands of tardigrades into orbit and exposed them directly to space vacuum and radiation for 10 days. When recovered, many had survived the experience and resumed normal activity. Some even reproduced successfully after returning to Earth, proving that space exposure hadn't damaged their reproductive systems. These experiments prove that life can survive space conditions, but they also reveal survival's limits. Exposure times matter enormously. Organisms that survive days or weeks in space show increasing damage over months or years. Even the toughest bacteria accumulate genetic mutations that eventually prove fatal. Survival rates decrease steadily with exposure duration. Protection becomes crucial for longer journeys. Most realistic panspermia scenarios involve organisms shielded inside meteorites, comets, or other natural spacecraft. Even a few centimeters of rock can block most cosmic radiation and provide thermal insulation against temperature extremes. But meteorites present their own challenges. During planetary impacts that launch rocks into space, materials experience acceleration forces exceeding 1 million times Earth's gravity. These forces generate shock waves and temperatures that can vaporize solid rock. Only organisms in specific locations within impact debris, protected by surrounding material and insulated from the worst thermal effects, have any chance of surviving the launch process. Computer simulations suggest that such survivable zones do exist. Deep within large impact fragments, temperatures might remain low enough and shock forces mild enough to preserve living organisms. The same rock that provides protection during space travel might also shield life during the violent process of ejection from its home planet. Water ice offers another form of protection. Comets contain vast quantities of frozen water that could shield embedded organisms from radiation while preserving them at extremely low temperatures. As comets approach the Sun and begin sublimating, they might release clouds of preserved microorganisms directly into planetary atmospheres. Recent research has revealed an unexpected source of natural radiation shielding: organic molecules themselves. Complex carbon compounds can absorb and dissipate radiation energy, protecting embedded organisms from genetic damage. This discovery suggests that meteorites rich in organic material might provide ideal environments for biological space travel. The organic compounds serve double duty, acting as both radiation shields and potential food sources for arriving organisms. A spore embedded in organic-rich meteorite material might survive the journey between planets and find itself in a nutrient-rich environment, perfect for resumed growth. Time remains the ultimate constraint. Even organisms with extraordinary radiation resistance accumulate damage over extended periods. A journey between nearby planets, lasting thousands of years, pushes biological survival systems to their limits. Interstellar travel, requiring millions of years, approaches the absolute boundaries of what any known organism can endure. But life continues to surprise us. Scientists have revived bacteria from salt crystals 250 million years old. Spores have been recovered from amber, preserved for 25 million years. Some organisms seem capable of surviving geological timescales through mechanisms we don't fully understand. These discoveries suggest that the boundaries of biological space travel might extend far beyond what current models predict. Perhaps the most resilient organisms, under the most favorable conditions, could survive even the longest interstellar journeys. Perhaps life is tougher than we dare imagine. The space survival experiments also reveal something profound about life's adaptability. The same mechanisms that allow organisms to survive cosmic radiation also help them thrive in Earth's most extreme environments. Life that can endure space might also colonize environments we once considered completely sterile: the upper atmospheres of planets, the surfaces of asteroids, the interiors of comets. This realization transforms our understanding of where life might exist. Instead of being confined to planetary surfaces with liquid water and moderate temperatures, life might inhabit a vast range of cosmic environments. Space itself might support ecosystems we're only beginning to imagine. The emerging picture from astrobiology research is cautiously optimistic. Space is indeed hostile to life, but not insurmountably so. Under the right conditions, with adequate protection, some organisms can survive journeys between worlds. The universe may not be as sterile as we once believed. These findings set the stage for examining specific mechanisms of biological space travel. If life can survive the journey, how might it actually travel between planets and star systems? The answer lies in the cosmic debris that connects all worlds. The meteorites, comets, and dust grains that carry more than just rock and ice through the void. On August 27, 1996, David McKay held a piece of Mars in his hands. The meteorite, designated ALH 84001, weighed just under 2 kilograms and looked utterly unremarkable. Grey, pitted, and roughly the size of a loaf of bread, it had sat unnoticed in Antarctic ice for 13,000 years before being discovered by meteorite hunters. But when McKay and his team at NASA's Johnson Space Center examined its interior under powerful microscopes, they found something that sent shockwaves through the scientific world. Tiny structures that looked remarkably like fossilized bacteria. The announcement made headlines across the globe. President Bill Clinton gave a televised address about the implications for humanity's place in the universe. Scientists debated the evidence with fierce intensity. Had we finally found proof of life beyond Earth? Were we looking at the remains of ancient Martian organisms that had traveled 4.5 billion kilometers to reach us? 25 years later, the debate continues, but ALH 84001 accomplished something more important than settling questions about Martian life. It demonstrated beyond doubt that rocks can travel between planets, carrying with them whatever microscopic passengers they might harbor. It proved that meteorites aren't just cosmic debris, but potential spacecraft capable of transporting life across the solar system. The journey of ALH 84001 began 15 million years ago, with a massive asteroid impact on Mars. The collision blasted millions of tons of Martian rock into space, hurling fragments at velocities exceeding Mars's escape velocity of 5 kilometers per second. Most of this debris fell back to the planet's surface or entered orbits around the Sun, but some pieces, including the rock that would become ALH 84001, found themselves on trajectories that would eventually intersect Earth's orbit. For 15 million years, this chunk of Mars drifted through space, following a complex orbital dance governed by gravitational interactions with planets, asteroids, and the Sun itself. Solar radiation gradually darkened its surface. Cosmic rays penetrated its interior, creating distinctive isotope signatures that would later help scientists identify its Martian origin. Micrometeorite impacts peppered its surface with tiny craters. Then, 13,000 years ago, Earth's gravity captured the wandering rock. It plunged through our planet's atmosphere at tremendous speed, heating to incandescence as air friction stripped away its outer layers. The surviving core, now considerably smaller than when it left Mars, crashed into the Antarctic ice sheet and began its long wait for discovery. ALH 84001 represents just one example of a process that has been occurring throughout solar system history. Computer models suggest that millions of rocks have traveled from Mars to Earth over the past 4 billion years. Hundreds of confirmed Martian meteorites now sit in laboratories around the world, each one a messenger from our planetary neighbor. But the traffic isn't one way. Earth rocks have also made the journey to Mars, though the physics make this transfer more challenging. Earth's stronger gravity requires higher impact velocities to launch material into space, and our dense atmosphere tends to slow down incoming projectiles that might cause such impacts. Nevertheless, calculations suggest that Earth material has occasionally reached Mars, Venus, and even more distant destinations. The implications for panspermia are profound. If life existed on early Mars when that planet possessed a thick atmosphere and liquid water on its surface, then Martian organisms could have been traveling to Earth for billions of years. Conversely, if life arose first on Earth, it could have seeded Mars during that planet's habitable period. The two worlds might have been exchanging biological material throughout their histories. But successful biological transport requires more than just getting rocks from one planet to another. The organisms inside must survive 3 distinct phases of their journey: ejection from their home planet, travel through space, and arrival at their destination. Each phase presents unique challenges that have shaped our understanding of how life might spread between worlds. The ejection phase is perhaps the most violent. When a large asteroid or comet strikes a planet, it releases energy equivalent to millions of nuclear weapons. The impact creates a crater many kilometers across and hurls material outward at velocities that can exceed 10 kilometers per second. Temperatures at the impact site reach thousands of degrees, hot enough to melt rock and vaporize water. Most material near the impact point gets sterilized instantly, but shockwave physics creates a complex pattern of pressures and temperatures throughout the impact zone. Computer simulations reveal that certain locations within the ejected material experience much milder conditions. Deep within large fragments, temperatures might remain low enough for organisms to survive, especially if those organisms exist in dormant states that can withstand brief temperature spikes. The key insight is that not all ejected material experiences the same conditions. A large impact creates a spray of debris with vastly different histories. Some fragments get completely melted and sterilized. Others experience moderate heating that might actually help preserve organisms by driving off water and creating a more stable environment for long-term storage. A few lucky pieces might escape with minimal heating, carrying their biological cargo intact into space. Laboratory experiments have tested whether organisms can survive impact-related shockwaves. Researchers use specially designed guns to accelerate projectiles containing bacteria and spores to velocities simulating planetary impacts. The results show that while most organisms die from the intense pressures and temperatures, some hardy species can survive impacts that generate pressures exceeding 50,000 times Earth's atmospheric pressure. These survival rates might seem low, but they become significant when multiplied by the enormous quantities of material ejected during major impacts. A single large collision might launch billions of rock fragments into space. Even if only a tiny fraction carry viable organisms, that still represents millions of potential biological spacecraft beginning their journeys to other worlds. The space travel phase presents different challenges. As we explored in previous chapters, organisms must survive radiation, temperature extremes, and vacuum conditions for thousands or millions of years. But meteorites provide natural protection that dramatically improves survival odds compared to naked exposure to space. Rock is an excellent radiation shield. A meteorite, just 1 meter thick, blocks most cosmic rays and solar particles. The interior of such a rock receives radiation doses only slightly higher than organisms experience on Earth's surface. For radiation-resistant bacteria and spores, this level of exposure poses minimal threat during journey times of thousands or even millions of years. Temperature control becomes more complex. The outer surface of a meteorite can experience wild temperature swings, from hundreds of degrees Celsius when facing the sun to near absolute zero in shadow. But rock is also an excellent insulator. Just centimeters beneath the surface, temperatures remain much more stable, buffered by the thermal mass of the surrounding stone. Water content plays a crucial role in temperature regulation. Many meteorites contain significant amounts of water locked in mineral crystals or trapped in tiny pockets. This water can act as a thermal buffer, absorbing heat during warm periods and releasing it during cold phases. It also provides a potential resource for any organisms that might become active during the journey. Some meteorites contain substantial amounts of organic compounds, the carbon-based molecules that form the building blocks of life. These organics can serve multiple functions during biological space travel. They provide radiation shielding, absorbing and dissipating high-energy particles before they reach embedded organisms. They offer potential nutrients for any microbes that might resume activity during the journey. And they create chemical environments that might actually enhance organism survival. The Murchison meteorite, which fell in Australia in 1969, contains over 90 different amino acids, many not found in terrestrial biology. It also harbors complex organic polymers, sugar derivatives, and even structures that resemble cell membranes. Such organic-rich meteorites could provide ideal environments for biological space travel, offering both protection and sustenance for hitchhiking organisms. Recent advances in analytical techniques have revealed just how common organic compounds are in meteorites. Nearly every carbon-rich meteorite examined contains complex organic molecules. Some harbor more organic material per unit mass than fertile Earth soil. These findings suggest that the raw materials for life are distributed throughout the solar system, carried by the same meteorites that might transport living organisms between planets. The arrival phase presents the final test for biological space travelers. Meteorites entering planetary atmospheres experience intense heating as air friction converts their orbital velocity into thermal energy. Surface temperatures can exceed 2,000 degrees Celsius, hot enough to melt most rocks, and certainly lethal to any surface-dwelling organisms. But once again, physics creates survivable niches within an overall deadly environment. The heating affects primarily the outer layers of incoming meteorites. Interior regions remain much cooler, protected by the surrounding rock and the brief duration of atmospheric passage. Large meteorites are particularly likely to preserve cool interior zones during entry. The size threshold for biological survival depends on entry velocity and atmospheric properties, but calculations suggest that meteorites larger than about 10 centimeters across can preserve interior regions cool enough for organism survival. Given that many meteorites reaching Earth's surface exceed this size, biological passengers have a reasonable chance of surviving the final phase of their interplanetary journey. Laboratory experiments have confirmed that organisms can survive simulated meteorite entries. Researchers have embedded bacteria and spores in rock samples, then subjected them to heating profiles that match those experienced by meteorites during atmospheric passage. Results show that organisms located more than a few centimeters from the surface can survive entry heating, especially if they exist in dormant states that resist thermal damage. These experiments also reveal the importance of water content and organic compounds in protecting biological passengers. Meteorites with higher water content provide better thermal buffering during entry heating. Those rich in organic compounds offer additional protection through their heat-absorbing properties. The same materials that make meteorites potentially habitable during space travel also help them deliver their biological cargo safely to planetary surfaces. But successful delivery requires more than just survival. Organisms must also find themselves in environments suitable for resumed activity after arrival. A spore that survives the entire journey from Mars to Earth accomplishes nothing if it lands in a sterile desert or toxic environment. The final step in successful panspermia requires favorable conditions for establishing new populations on alien worlds. Earth provides numerous examples of environments where meteorite-delivered organisms might thrive. Impact craters often contain water from groundwater or rainfall. The heat of impact can create hydrothermal systems that persist for years or decades after the initial collision. The pulverized rock around impact sites often contains nutrients that could support microbial growth. Some impact craters become lakes or wetlands, providing ideal conditions for aquatic microorganisms. Others create unique geological formations that might serve as protective habitats for extremophile organisms. The Vredefort crater in South Africa, created by an impact 2 billion years ago, still shows evidence of the hydrothermal systems that followed the collision. Such environments could have provided perfect conditions for Martian organisms to establish themselves on Earth. The organic compounds delivered by meteorites might also create favorable chemical environments for arriving organisms. Impact events can produce a rich soup of organic molecules through shock chemistry, creating conditions that might actually be more favorable for life than the original planetary environments. Some scientists speculate that impacts might not just deliver life, but also create new ecological niches that enhance its chances of successful establishment. Modern meteorite falls continue to provide insights into biological transport mechanisms. The Tagish Lake meteorite, which fell in Canada in 2000, was collected so quickly that researchers could analyze its organic content before terrestrial contamination occurred. They found a complex mixture of organic compounds including amino acids, nucleotide bases, and even structures resembling primitive cell membranes. More intriguingly, some meteorites contain what appear to be fossilized microstructures that resemble known microorganisms. These structures, found in several different meteorite types, show organized patterns and chemical compositions consistent with biological origin. While their interpretation remains controversial, they suggest that meteorites might indeed carry the remains of ancient life forms. The search for active, viable organisms in meteorites continues to challenge researchers. Distinguishing between indigenous meteoritic life and terrestrial contamination requires extraordinary care and sophisticated analytical techniques. But several research groups claim to have found living microorganisms in freshly fallen meteorites, organisms with biochemical signatures suggesting non-terrestrial origin. These claims remain highly controversial and require independent verification before acceptance by the broader scientific community. But they highlight the possibility that meteorites might still be delivering living organisms to Earth today, continuing a process that has operated throughout solar system history. The cumulative evidence from meteorite research paints a compelling picture of biological transport possibilities. Rocks can and do travel between planets, carrying with them whatever microscopic passengers they might harbor. The physics of impact ejection, space travel, and atmospheric entry all allow for organism survival under specific conditions. The chemical environments within many meteorites provide protection and potentially even nutrients for biological space travelers. These findings transform meteorites from simple chunks of cosmic debris into sophisticated biological transport vehicles. Each meteorite that reaches Earth represents a potential capsule from another world, carrying information about the conditions, chemistry, and possibly even the life of its origin planet. The implications extend far beyond our solar system, suggesting that similar processes might operate around every star in the galaxy. As we prepare to examine specific evidence from Mars and its potential role as life's first home in our solar system, we must remember that meteorites provide more than just theoretical possibilities for biological transport. They offer tangible, testable evidence that life might indeed travel between worlds on ships of stone, connecting planetary biospheres in ways we're only beginning to understand. 4 billion years ago, two worlds in our solar system harbored conditions suitable for life. One was Earth, still cooling from its violent birth, but already developing the first hints of an atmosphere and liquid water oceans. The other was Mars, a planet that bore little resemblance to the cold, arid world we know today. Ancient Mars possessed something modern Mars has lost: hope. Picture Mars as it existed in those distant days. Rivers carved intricate networks across its surface, flowing toward vast lakes and perhaps even shallow seas. An atmosphere thick enough to support liquid water protected the planet's surface from the harsh radiation of space. Volcanic activity pumped gases into the air and created hydrothermal systems where chemistry could experiment with increasing complexity. For perhaps 1 billion years, Mars offered conditions as favorable for life as those found on early Earth. This ancient Mars has emerged from decades of robotic exploration that revolutionized our understanding of planetary evolution. The story began with grainy images from Mariner spacecraft in the 1960s, continued through the detailed mapping by orbiting satellites, and reached new heights with rovers that have spent years crawling across the Martian surface, analyzing rocks and soil with instruments more sophisticated than many Earth-based laboratories. Each mission revealed new evidence for ancient water activity. Orbital photographs showed river valleys carved by flowing water, deltas where streams emptied into ancient lakes, and mineral deposits that could only form in the presence of liquid water. Rovers examined rocks that clearly formed in aquatic environments, some containing the chemical signatures of hot springs or underground water systems. The Mars Reconnaissance Orbiter discovered something even more tantalizing: recurring slope lineae, dark streaks that appear and disappear seasonally on Martian slopes. These features might represent briny water flowing on the surface of modern Mars, suggesting that liquid water activity continues even today under specific conditions. If water still flows on Mars, then the planet might retain some of its ancient habitability. But the most compelling evidence for ancient Martian habitability comes from the Curiosity rover's exploration of Gale Crater. This 96-mile-wide impact basin once contained a lake that persisted for millions of years. The rover analysed sediment layers that formed on the lake bottom, finding complex organic molecules and mineral assemblages that indicate pH-neutral water suitable for life. Curiosity's instruments detected sulfur, nitrogen, hydrogen, oxygen, phosphorus and carbon in the ancient lake sediments. These 6 elements form the foundation of all known life. The rover also found minerals like hematite and goethite that form in oxygenated water, suggesting that early Mars might have possessed free oxygen in its atmosphere or water systems. The Perseverance rover, currently exploring Jezero Crater, has extended these discoveries. Jezero once hosted a river delta where flowing water deposited sediments rich in organic compounds. The rover has collected samples that show clear evidence of interaction between water and rock over extended periods, creating chemical environments that could have supported microbial communities. These robotic explorers have painted a picture of ancient Mars that bears striking resemblances to early Earth. Both planets possessed liquid water, complex chemistry, and energy sources that could drive biological processes. Both experienced volcanic activity that created diverse mineral environments. Both developed atmospheric compositions that could support surface water and protect against harmful radiation. But Mars possessed advantages that early Earth lacked. The red planet's smaller size meant it cooled faster after formation, potentially becoming habitable millions or even hundreds of millions of years before Earth achieved similar conditions. Mars also experienced less intense bombardment during the late heavy bombardment period that sterilized much of the inner solar system around 4 billion years ago. These timing differences have profound implications for life's origin. If Mars became habitable first and remained suitable for life longer than previously thought, then life might have arisen there before spreading to Earth through meteorite transfer. We might all be descendants of ancient Martians, carried to Earth by the same rock transport mechanisms that continue to operate today. The case for Martian origin becomes more compelling when we consider the chemical environments that rovers have discovered. Gale Crater's ancient lake contained dissolved minerals that would have provided ideal conditions for certain types of Earth bacteria. The pH levels, salinity, and chemical composition match closely with environments where extremophile organisms thrive on Earth today. Some of these Martian environments might have been even more favorable for life's origin than anything available on early Earth. The planet's lower gravity would have allowed taller volcanic structures and deeper impact craters, creating more diverse geological settings. The thinner atmosphere would have permitted more efficient chemical cycling between surface and atmospheric reservoirs. Mars also possessed abundant clay minerals that could have served as templates for organizing complex organic molecules. These clays, detected by multiple rovers and orbiting instruments, form in the presence of liquid water and can concentrate and organize biological precursors. Some scientists suggest that such mineral surfaces might have been essential for life's origin, providing the scaffolding needed to assemble the first self-replicating molecules. The planet's magnetic field, though weaker than Earth's, could have provided some protection against cosmic radiation during life's most vulnerable early stages. Localized magnetic anomalies detected by orbiting spacecraft suggest that ancient Mars possessed a more complex and perhaps stronger magnetic field than exists today. But perhaps the most intriguing evidence for possible Martian life comes not from the planet's surface, but from the meteorites it has sent to Earth. Over 100 confirmed Martian meteorites now sit in laboratories worldwide, each one a time capsule preserving information about conditions on ancient Mars. Some of these meteorites contain organic compounds that are difficult to explain through purely geological processes. The Nebula meteorite, which fell in Egypt in 1911, harbors complex carbon molecules and mineral assemblages that suggest interaction with Martian groundwater. The organic compounds show isotopic ratios that differ from typical terrestrial or meteoritic organics, hinting at possible biological origin. But it's the ALH 84001 meteorite that continues to generate the most intense scientific debate. Beyond the microscopic structures that might represent fossilized bacteria, this rock contains other features suggestive of biological activity. Magnetite crystals within the meteorite resemble those produced by certain Earth bacteria. Organic compounds are associated with carbonate minerals in patterns that might indicate biological processing. The structures themselves remain highly controversial. They are much smaller than typical Earth bacteria, leading some scientists to dismiss them as mineral artifacts rather than biological fossils. Others argue that the structures are too organized and too consistently shaped to be purely geological. The debate has continued for over 25 years without resolution, highlighting both the difficulty of recognizing ancient life and the extraordinary claims that require extraordinary evidence. More recent analysis of ALH 84001 using advanced techniques has revealed additional intriguing features. Electron microscopy shows that some of the organic compounds are organized in ways that resemble biological membranes. Chemical analysis indicates that the organic materials formed under temperature and pressure conditions consistent with biological activity rather than high-temperature geological processes. Other Martian meteorites have yielded similar puzzling features. The Chagoti meteorite contains organic compounds and mineral textures that some researchers interpret as evidence of biological activity. The Tissant meteorite, which fell in Morocco in 2011 and was collected quickly enough to avoid terrestrial contamination, harbors complex organic molecules that are difficult to explain through known abiotic processes. These meteoritic findings remain far from conclusive evidence for Martian life. Each claimed biosignature has alternative explanations involving purely chemical processes. The organic compounds might result from interactions between minerals and carbon-bearing fluids. The microscopic structures might represent unusual crystal growth patterns. The chemical compositions might reflect unknown aspects of Martian geology. But taken together, these features paint a suggestive picture. Ancient Mars clearly possessed conditions suitable for life. Martian meteorites contain organic compounds and structures that are at least consistent with biological activity. The timing of Mars's habitable period coincides with the proposed origin of life in our solar system. The planet's geological history includes environments that match closely with those where life thrives on Earth today. The implications extend beyond simple questions of whether life existed on ancient Mars. If Martian life arose independently from terrestrial life, then biology represents a predictable outcome of planetary evolution rather than a unique accident. If Martian and terrestrial life share common ancestry through meteorite exchange, then life might be a solar system-wide phenomenon that transcends individual planetary boundaries. Recent discoveries have added new dimensions to the Martian life debate. The detection of methane in Mars's atmosphere by multiple spacecraft and rovers suggests ongoing biological or geological activity. On Earth, most atmospheric methane is produced by living organisms, though geological processes can also generate this gas. The seasonal variations in Martian methane concentrations might indicate active biological processes responding to changing environmental conditions. Perchlorate compounds detected in Martian soil initially seemed to rule out surface life by creating conditions toxic to most known organisms. But subsequent research revealed that some Earth bacteria can actually use perchlorates as energy sources, thriving in environments that would kill other life forms. Mars's perchlorate-rich soil might not be a barrier to life, but rather a unique ecological niche that could support specialized organisms. Subsurface water discoveries have expanded the potential habitable zones on modern Mars. Radar observations from orbiting spacecraft reveal extensive groundwater systems beneath the planet's surface, including possible liquid water lakes beneath the polar ice caps. These subsurface environments might provide stable habitats where Martian life could persist despite surface conditions that seem inhospitable. The search for current Martian life has intensified with each new discovery. The Perseverance rover is specifically designed to look for biosignatures in ancient rocks, using instruments that can detect organic compounds, analyze mineral compositions, and even search for microscopic structures that might represent fossilized life. The samples it collects are being cached for eventual return to Earth, where they can be analyzed with laboratory instruments far more sophisticated than anything that can be sent to Mars. Future missions will extend this search in unprecedented ways. The Mars Sample Return mission, planned for the late 2020s, will bring Perseverance's samples back to Earth for definitive analysis. The European Space Agency's ExoMars rover will drill up to 2 meters beneath the surface, searching for organic compounds and possible biosignatures in subsurface environments protected from surface radiation. These missions might finally answer the question that has tantalized scientists for decades. Did life ever exist on Mars? But even if they find definitive evidence for ancient or current Martian biology, deeper questions will remain. Did Martian life arise independently, or does it share ancestry with terrestrial organisms through meteorite exchange? If life exists on Mars today, is it related to ancient Earth life that colonized Mars? Or does it represent a completely separate genesis? The answers to these questions will reshape our understanding of life's place in the universe. If Mars harbored independent biology, then life represents a common outcome of planetary evolution, likely to arise wherever conditions permit. If Martian and terrestrial life share common ancestry, then individual planets are not isolated biological islands, but part of larger living systems that span entire solar systems. Either possibility suggests that life might be far more common in the universe than we currently recognize. Every star system with multiple planets might experience biological exchange similar to what occurred between Earth and Mars. Every habitable world might be connected to others through the same meteorite transport mechanisms that link our planet to its red neighbor. As we prepare to explore even more exotic possibilities for life's cosmic distribution, we must remember that Mars represents our best current example of how biology might spread between worlds. Whether as life's original home or as Earth's biological colony, Mars demonstrates that habitability transcends individual planetary boundaries. The red planet teaches us that life, once established, might be far more mobile and persistent than we ever imagined, traveling between worlds on ships of stone and establishing itself wherever conditions permit growth and reproduction. In November 2014, humanity achieved something unprecedented in the 4.5 billion year history of our solar system. For the first time, we gently landed a spacecraft on a comet and began to examine its interior composition with scientific instruments. The Philae lander, carried to comet 67 P. Churyumov-Gerasimenko by the European Space Agency's Rosetta mission, bounced twice before settling into a shadowed depression on the comet's surface. Over the following days, It transmitted data that revolutionized our understanding of these ancient wanderers and their potential role in spreading life throughout the cosmos. What Philae found challenged every assumption about comets being simple, dirty snowballs. The comet's interior harbored a complex chemistry that included 16 different organic compounds, some containing nitrogen and phosphorus essential for biological molecules. Water ice was present, but in forms and concentrations that suggested a far more dynamic history than scientists had expected. Most intriguingly, the comet showed evidence of internal chemical processes that might have operated for millions of years during its formation in the outer solar system. This single mission transformed comets from peripheral players in panspermia theories into potential starring roles. If comet Churyumov-Gerasimenko, a relatively typical short-period comet, contains such complex organic chemistry, then the trillions of comets in our solar system might represent vast reservoirs of prebiotic compounds and possibly even living organisms preserved in cosmic deep freeze. Comets form in the coldest, most distant regions of planetary systems, where temperatures hover just a few degrees above absolute zero. In our solar system, they originate primarily in two regions: the Kuiper Belt, extending from Neptune's orbit to roughly 50 times Earth's distance from the Sun, and the Oort cloud, a vast spherical shell of icy bodies that may extend halfway to the nearest star. These formation environments provide ideal conditions for preserving complex organic molecules and potentially even living organisms. The extreme cold essentially stops all chemical reactions, creating perfect preservation conditions that can last for billions of years. Any biological material incorporated into comets during their formation would remain unchanged throughout their long storage in the outer solar system. But comets don't remain in cold storage forever. Gravitational perturbations from passing stars, planetary migrations, or interactions with giant molecular clouds can destabilize comet orbits, sending them on journeys toward the inner solar system. As they approach the Sun, solar heating begins to sublimate their icy surfaces, creating the spectacular tails that have fascinated humans throughout history. This sublimation process might serve as a delivery mechanism for biological material. As comet ice vaporizes, it releases any embedded organisms into space, where they could potentially seed nearby planets. The process is gentler than the violent impacts that launch meteorites between planets, potentially offering better survival prospects for delicate biological structures. The Stardust mission, which collected samples from Comet Wild 2 in 2004, provided the first direct analysis of comet particles in terrestrial laboratories. The samples revealed an unexpectedly complex mixture of materials, including crystalline silicates that must have formed at high temperatures near the Sun before being incorporated into the comet in the cold outer solar system. More significantly, the Stardust samples contained glycine, the simplest amino acid, and a fundamental building block of proteins. This marked the first confirmed detection of an amino acid in a comet, proving that these ancient objects carry the chemical precursors necessary for life. Subsequent analysis revealed additional organic compounds, including complex carbon molecules that could serve as raw materials for biological systems. The Deep Impact mission, which deliberately crashed a projectile into Comet Tempel 1 in 2005, revealed even more about comet interior chemistry. The impact excavated material from deep within the comet's nucleus, exposing regions that had been protected from space weathering for billions of years. Spectroscopic analysis of the impact plume showed water vapor, carbon dioxide, and numerous organic compounds streaming away from the collision site. But perhaps the most significant comet research has come from ground-based observations of long-period comets arriving from the Oort cloud. These objects, which take thousands or millions of years to complete their orbits, preserve materials from the earliest days of solar system formation. They offer glimpses into the chemical conditions that existed when planets were first taking shape. Observations of Comet Hale-Bopp during its spectacular 1997 appearance revealed over 24 different molecules in its coma, the cloud of gas and dust that surrounds active comet nuclei. These included water, carbon monoxide, methanol, hydrogen cyanide, and numerous organic compounds. The detection of such molecular diversity suggested that comets might serve as vast chemical laboratories where complex organic synthesis could occur over geological timescales. More recent observations have pushed these discoveries even further. Comet Lovejoy, observed during its close approach to the Sun in 2011, was found to release ethyl alcohol and sugar derivatives as its nucleus heated up. The quantities were staggering. The comet was producing the equivalent of 500 bottles of wine per second at the peak of its activity. If comets can produce and preserve such complex organic molecules, they might also harbor the conditions necessary for life itself. The chemical complexity of comets suggests formation mechanisms more sophisticated than simple accumulation of ice and dust. Computer models now indicate that comet formation involved multiple phases of heating and cooling, chemical processing, and organic synthesis. During their formation in the outer solar system, comets might have experienced conditions favorable for the development of increasingly complex organic chemistry. Some researchers have proposed that comets might even support active biological processes. The interior of a large comet nucleus could remain above the freezing point of water for millions of years due to radioactive heating from incorporated rock and metal. This could create liquid water environments where biological processes might operate, even in the cold outer solar system. Laboratory experiments have tested whether organisms could survive conditions inside comet nuclei. Bacteria and spores exposed to simulated comet environments have shown remarkable resilience, surviving freeze-thaw cycles, radiation exposure, and the chemical conditions expected in comet interiors. Some organisms not only survived, but actually showed signs of metabolic activity when provided with the organic compounds commonly found in comets. These findings suggest that comets might serve as more than just passive carriers of biological material. They might provide active environments where life could persist and possibly even evolve during long journeys between star systems. A comet ejected from one stellar system and captured by another could carry living ecosystems across interstellar distances, seeding new worlds with fully developed biological communities. The delivery mechanism for comet-based panspermia operates differently from meteorite transport. Instead of violent impacts that might sterilize biological cargo, comets could gently deposit organisms into planetary atmospheres through their natural outgassing processes. As a comet approaches a star, solar heating creates jets of gas and dust that stream away from the nucleus. These jets could carry viable organisms directly into the atmospheres of nearby planets. The process becomes even more intriguing when we consider that comet tails can extend for millions of kilometers. A comet passing through a planetary system could essentially paint its biological contents across vast volumes of space, increasing the probability that some organisms would encounter habitable worlds. The particles in comet tails travel at relatively low velocities compared to meteorite impacts, potentially allowing for gentler deliveries that preserve biological integrity. Recent research has revealed that our solar system is constantly bombarded with microscopic particles from interstellar space. The Ulysses and Galileo spacecraft detected streams of dust grains entering our solar system from the direction of the local interstellar cloud, the region of space through which our Sun is currently moving. Some of these particles might originate from comets in other star systems, carrying biological material across interstellar distances. The implications are staggering. If comet-derived particles regularly cross interstellar space, then panspermia might operate not just within individual stellar systems, but across the entire galaxy. Every star system might be connected to every other through streams of microscopic particles, carrying organic compounds and possibly even living organisms. The galaxy itself might function as a vast biological network, with comets serving as the primary transport mechanism. This interstellar particle exchange could explain some puzzling observations about the distribution of organic compounds in space. Radio telescopes have detected over 200 different molecules in interstellar gas clouds, including complex organic compounds that are difficult to synthesize through purely chemical processes. Some of these molecules might originate from comet-related processes in distant stellar systems, spreading throughout the galaxy via microscopic dust grains. The Spitzer Space Telescope has observed organic compounds in the atmospheres of exoplanets hundreds of light years away. Some of these molecules are identical to compounds found in solar system comets, suggesting that comet chemistry might be universal throughout the galaxy. If comets everywhere produce similar organic inventories, then the basic chemical requirements for life might be distributed throughout cosmic space. But interstellar biological transport faces enormous challenges. The journey times between stars stretch to millions or billions of years, far exceeding the survival limits of any known organisms. Radiation levels in interstellar space are much higher than within stellar systems, where magnetic fields provide some protection. Temperature conditions approach absolute zero, and the density of matter is so low that meaningful chemical interactions become virtually impossible. However, recent discoveries about extremophile organisms have expanded our understanding of biological survival limits. Some bacteria can remain viable after millions of years in dormant states. Others show extraordinary resistance to radiation and temperature extremes. A few species seem capable of surviving conditions that approach those found in interstellar space. Laboratory experiments have begun testing whether organisms can survive simulated interstellar journeys. Bacteria and spores subjected to extreme cold, high radiation, and vacuum conditions for extended periods have shown surprising resilience. While survival rates decrease with exposure time, some organisms remain viable after treatments designed to simulate millions of years of interstellar travel. These experiments suggest that interstellar panspermia, while challenging, might not be impossible. If even a tiny fraction of organisms can survive interstellar journeys, and if comet-related processes distribute trillions of particles throughout the galaxy, then biological exchange between star systems might occur regularly over cosmic timescales. The discovery of organic compounds in interstellar dust grains supports this possibility. Analysis of particles collected in Earth's upper atmosphere has revealed complex organic molecules that appear to originate from interstellar space. Some show isotopic signatures that differ from solar system materials, confirming their extrasolar origin. While these particles don't contain recognizable biological structures, they demonstrate that organic material regularly crosses interstellar distances. Advances in analytical techniques are revealing ever greater complexity in cosmic dust chemistry. Particles collected from comet tails show layered structures that suggest multiple episodes of organic synthesis and processing. Some contain mineral phases that could only form in the presence of liquid water, indicating that comet interiors might experience much more dynamic chemistry than previously recognized. The Hayabusa missions, which returned samples from asteroids Itokawa and Ryugu, have extended our understanding of small body chemistry. While asteroids differ from comets in their formation environments and compositions, both types of objects show evidence of complex organic processing. The boundary between asteroids and comets becomes blurred when we consider their potential roles in biological transport. Some objects in our solar system appear to be hybrids, showing characteristics of both asteroids and comets. These transition objects might represent ideal vehicles for biological transport, combining the organic richness of comets with the structural stability of asteroids. They could serve as robust spacecraft capable of carrying biological cargo across vast distances while providing both chemical resources and physical protection. As we prepare to examine the most speculative aspect of panspermia, directed seeding by advanced civilizations, we must recognize that comets and cosmic dust represent the most plausible mechanisms for spreading life across cosmic distances. Their vast numbers, complex chemistry, and gentle delivery mechanisms make them ideal candidates for biological transport, whether operating within individual stellar systems or across interstellar space. These ancient wanderers might serve as nature's own biological spacecraft, carrying the seeds of life wherever gravity and chance allow them to travel. Francis Crick was dying. The man who had unlocked the secret of life itself, co-discoverer of DNA's double helix structure, faced the ultimate biological mystery, his own mortality. But even as cancer consumed his body in 2004, his mind remained focused on an idea that had captivated him for decades. What if life on Earth wasn't a natural accident but an artificial creation? What if we are all products of the most ambitious engineering project ever undertaken? Crick had first proposed directed panspermia in 1973, collaborating with chemist Leslie Orgel to present what seemed like science fiction masquerading as serious science. They suggested that an advanced alien civilization had deliberately seeded Earth with life billions of years ago, launching specially designed organisms across interstellar space to colonize suitable planets throughout the galaxy. The proposal seemed outrageous, the kind of speculation that serious scientists avoided for fear of damaging their reputations. But Crick was no ordinary scientist. His credentials were impeccable, his analytical mind legendary, and his willingness to consider radical possibilities had already revolutionized biology once. If Francis Crick thought directed panspermia deserved serious consideration, then perhaps it did. The logic behind directed panspermia emerged from problems that haunted conventional theories of life's origin. How could the extraordinary complexity of even the simplest living cells arise from random chemical processes? Why did all terrestrial life use the same genetic code when thousands of alternatives seemed equally viable? Why did life appear so quickly in Earth's geological record, leaving little time for the gradual chemical evolution most theories required? Crick recognized that these problems might have solutions that didn't involve natural processes at all. An advanced civilization could have solved the complexity problem through intelligent design, creating organisms specifically engineered for survival and reproduction. The universal genetic code might reflect the biochemical preferences of alien designers rather than chemical inevitability. Life's rapid appearance on Earth might result from deliberate introduction rather than spontaneous generation. The idea gained respectability through Crick's careful scientific reasoning. He calculated the energy requirements for launching biological payloads across interstellar distances, concluding that a sufficiently advanced civilization could indeed accomplish such a feat. He analyzed the types of organisms that might survive interstellar journeys, suggesting that specially designed microbes could be engineered for extreme longevity and radiation resistance. Crick and Orgel proposed that the seeding civilization might have used automated spacecraft carrying frozen or dormant organisms. These biological cargo ships could travel for millions of years before reaching suitable target systems, where they would deploy their living payloads on promising planets. The process could be repeated throughout the galaxy, creating vast networks of related life forms spanning hundreds or thousands of star systems. But why would any civilization undertake such an enormous project? Crick suggested several possible motivations. Perhaps the seeding species faced extinction and wanted to preserve life in some form throughout the galaxy. Perhaps they viewed seeding as a moral imperative, spreading the gift of consciousness to otherwise sterile worlds. Perhaps they conducted it as a vast scientific experiment, studying how life evolved under different planetary conditions. The hypothesis also explains some puzzling features of terrestrial biology. All Earth life depends on molybdenum for certain essential enzymes, despite this element's rarity in Earth's crust. Molybdenum is much more abundant around certain types of stars, suggesting that our biological chemistry might have originated elsewhere and been transported to Earth intact. Similarly, life's apparent preference for specific molecular forms over their chemical mirror images might reflect the biochemical quirks of alien designers rather than fundamental physical principles. The fact that Earth organisms use only 20 amino acids out of hundreds of possibilities might indicate artificial selection rather than natural optimization. These observations remained highly speculative, and critics pointed out that natural processes could also explain most of them. But directed panspermia offered a coherent alternative framework that addressed multiple puzzles simultaneously. Even if unprovable, it provided a useful thought experiment that illuminated assumptions underlying conventional theories. The concept evolved and diversified as other researchers adopted and modified Crick's basic idea. Some proposed that seeding might be an ongoing process, with advanced civilizations continuously launching biological missions throughout their lifespans. Others suggested that seeding might occur accidentally, with civilizations inadvertently spreading their own microbes during interstellar exploration. The astronomer Thomas Gold proposed that directed panspermia might explain the origin of life's chirality, its preferential use of left-handed amino acids and right-handed sugars. This molecular handedness appears arbitrary from a chemical perspective, but is universal in terrestrial biology. Gold suggested that alien life might have established this preference, which was then transmitted to all subsequently seeded worlds. Carl Sagan, initially skeptical of directed panspermia, eventually acknowledged its theoretical legitimacy while emphasizing the extraordinary evidence that would be required to support such claims. He suggested that definitive proof might come from discovering artificial signatures embedded in the genetic code itself, perhaps in the form of mathematical patterns or symbolic messages that could only result from intelligent design. The search for such genetic messages became a cottage industry among researchers intrigued by directed panspermia possibilities. Some claimed to find meaningful patterns in DNA sequences, mathematical constants, encoded in protein structures, or symbolic representations hidden in the genetic code. Most of these discoveries proved to be statistical artifacts or wishful thinking, but the searches continued as analytical techniques became more sophisticated. Meanwhile, advances in astrobiology began to make directed panspermia seem less far-fetched. The discovery of extremophile organisms that could survive conditions previously thought impossible for life expanded the range of organisms that might be suitable for interstellar transport. Advances in genetic engineering demonstrated that organisms could indeed be artificially modified to survive extreme environments. The development of synthetic biology took these possibilities even further. Scientists began creating entirely artificial organisms designed for specific purposes, demonstrating that life could indeed be engineered rather than merely discovered. If human scientists could create custom organisms with desired properties, then advanced alien civilizations might possess capabilities that dwarfed our current achievements. Projects like Craig Venter's creation of synthetic bacterial genomes showed that artificial life was not only possible, but practical. Venter's team constructed complete bacterial chromosomes from scratch, incorporating synthetic genes and artificial regulatory systems. They even embedded coded messages in the genome, including email addresses and famous quotations, proving that DNA could indeed carry non-biological information. These achievements suggested that directed panspermia might not require exotic technologies beyond the reach of biological civilizations. A species with genetic engineering capabilities only moderately more advanced than our current techniques, could theoretically design organisms capable of surviving interstellar journeys and establishing themselves on alien worlds. The spacecraft required for directed panspermia also became more plausible as space technology advanced. Concepts for interstellar probes powered by fusion rockets, antimatter engines, or even light sails showed that unmanned missions to nearby star systems lay within the realm of theoretical possibility. A civilization with sufficient motivation and resources could launch biological seeding missions using technologies that weren't completely beyond imagination. But directed panspermia faced the same survival challenges that limited natural panspermia. Even artificially enhanced organisms would struggle to survive interstellar journeys lasting millions of years. Radiation damage, metabolic decay, and simple time would exact their toll on any biological system, no matter how cleverly designed. Some researchers propose solutions involving more exotic technologies. Perhaps seeding civilizations use suspended animation techniques that could preserve organisms indefinitely. Perhaps they employed nanotechnology to repair radiation damage during flight. Perhaps they developed completely artificial life forms that could survive conditions impossible for natural organisms. These speculations pushed directed panspermia into realms that were difficult to distinguish from science fiction. Without concrete evidence for such technologies, the hypothesis remained largely untestable. Critics argued that invoking alien intelligence to explain biological puzzles violated scientific principles by introducing entities that could neither be observed nor measured. Yet the criticism missed an important point. Directed panspermia wasn't necessarily intended as a definitive explanation for life's origin, but rather as a framework for thinking about alternative possibilities. By considering what evidence would support artificial seeding versus natural processes, scientists could design better tests to distinguish between different hypotheses. The hypothesis also raised profound philosophical questions about humanity's place in the universe. If we are products of alien engineering, what does that mean for our understanding of consciousness, free will, and moral responsibility? Are we obligated to continue the seeding process ourselves, spreading life to other worlds as our creators might have intended? Do we have the right to modify or enhance the biological inheritance we received from alien benefactors? These questions became more pressing as human space exploration capabilities advanced. Several organizations began seriously planning missions to Mars, Europa, and other potentially habitable worlds in our solar system. The scientific community grappled with planetary protection protocols designed to prevent contamination of pristine environments with Earth organisms. But what if those environments were already contaminated by previous seeding missions? What if Mars harbored descendants of the same biological lineage that seeded Earth? The discovery of related organisms on multiple worlds might provide evidence for directed panspermia, but it could also complicate efforts to distinguish between natural and artificial distribution mechanisms. Some researchers propose that human civilization itself might represent evidence for directed panspermia's success. Our rapid technological development, curiosity about other worlds, and growing capabilities for space exploration might reflect intended outcomes of an alien seeding project. Perhaps we are becoming the very type of civilization capable of continuing the seeding process ourselves. This perspective transformed directed panspermia from a historical hypothesis about past events into a predictive framework about future possibilities. If advanced civilizations naturally develop seeding capabilities and motivations, then we might expect to observe ongoing seeding activities in our galaxy. The universe might be filled with biological missions launched by countless civilizations throughout cosmic history. The search for evidence of such activities has become a legitimate scientific endeavor. Radio telescopes scan the sky for artificial signals that might indicate active seeding missions. Space-based observatories look for unusual objects that might represent alien spacecraft or biological cargo containers. Analyses of meteorites and cosmic dust search for artificial signatures that might indicate intelligent origin. So far, these searches have found no conclusive evidence for ongoing directed panspermia activities. But the absence of evidence is not evidence of absence, particularly when dealing with phenomena that might operate on timescales exceeding human observation capabilities. A galaxy-wide seeding program might unfold over millions or billions of years, making individual events extremely rare from our limited temporal perspective. The advent of powerful space telescopes and sophisticated analytical techniques has begun to make directed panspermia testable in ways that seemed impossible when Crick first proposed the idea. Future missions to other worlds in our solar system might discover biological material that shows clear signs of artificial modification. Analysis of extremophile organisms on Earth might reveal genetic features that are difficult to explain through natural evolution. The discovery of technological civilizations around other stars would transform directed panspermia from speculation into serious scientific hypothesis. If we find evidence that intelligence commonly arises in planetary systems, then biological seeding becomes a plausible activity that such civilizations might pursue. The universe might be filled with the descendants of countless seeding missions, creating vast biological networks that connect star systems across the galaxy. As we prepare to examine the evidence we currently have for life's cosmic connections, we must remember that directed panspermia represents more than just an exotic alternative to natural origin theories. It embodies humanity's growing recognition that we might not be alone in the universe and that intelligence might play a far greater role in cosmic evolution than we previously imagined. Whether or not aliens seeded Earth with life, the possibility forces us to think more broadly about life's place in the universe and our own responsibilities as a potentially seeding species ourselves. Science is not about belief. It is not about elegant theories or compelling narratives or profound philosophical implications. Science is about evidence, the slow accumulation of facts that either support or contradict our hypotheses about how the universe works. After decades of speculation about panspermia, after countless experiments and observations, after theoretical frameworks that range from plausible to fantastical, we must confront a fundamental question. What does the evidence actually tell us? The answer is both frustrating and fascinating. The evidence is suggestive but inconclusive, tantalizing but incomplete. Like a detective story with crucial pages missing, the case for panspermia rests on circumstantial evidence, intriguing clues, and logical deductions rather than smoking gun-proof. Yet this incomplete picture has emerged from some of the most sophisticated scientific investigations ever undertaken, revealing truths about life's history that were unimaginable just decades ago. The fossil record provides our deepest window into life's ancient history, preserving traces of organisms that lived billions of years ago when Earth was a radically different world. These fossilized remains tell a story that has surprised scientists with its implications for how quickly and how early life appeared on our planet. The oldest confirmed fossils date to approximately 3.5 billion years ago, preserved in ancient rock formations in Australia and South Africa. These structures, called stromatolites, were created by communities of cyanobacteria that built layered mats in shallow water environments. But even more intriguing are potential biosignatures that push life's origin back much earlier. In 2016, researchers announced the discovery of what might be fossilized microorganisms in rocks from Greenland, dating to 3.7 billion years ago. These structures, if genuinely biological, would represent the oldest direct evidence of life on Earth. Other studies have identified chemical signatures in rocks up to 4.1 billion years old that might indicate biological activity, though these claims remain highly controversial. The significance of these dates becomes clear when we consider Earth's early history. 4.6 billion years ago, our planet formed from the collision and accretion of countless smaller bodies. For several hundred million years afterward, Earth remained largely molten, bombarded by asteroids and comets in the chaotic aftermath of planetary formation. The surface began to cool and liquid water oceans formed perhaps 4.4 billion years ago. This means that life appeared on Earth within just a few hundred million years after conditions became potentially habitable, possibly much sooner. In geological terms, this represents an extraordinarily rapid transition from sterile rock to thriving biological communities. The speed of this transition has puzzled scientists for decades and remains one of the strongest arguments in favor of panspermia hypotheses. The rapidity becomes even more remarkable when we consider the complexity of even the earliest life forms. The fossilized cyanobacteria from 3.5 billion years ago were already sophisticated organisms capable of photosynthesis, one of biology's most complex biochemical processes. They possess cell walls, internal structures, and metabolic pathways that rival those of modern bacteria in their intricacy. This level of complexity suggests a long evolutionary history preceding the formation of these earliest fossils. If life required hundreds of millions of years to evolve from simple chemicals to photosynthetic bacteria, then its origin must have occurred almost immediately after Earth became habitable, or perhaps even before our planet was ready to support such organisms. The genetic evidence provides another perspective on life's early history, one that both supports and complicates panspermia hypotheses. Modern molecular biology has revealed that all known life shares fundamental biochemical features that suggest common ancestry. The genetic code, the structure of cellular membranes, and basic metabolic pathways are essentially identical across all domains of life, from bacteria to humans. This universality could support panspermia by suggesting that all terrestrial life descended from a single seeding event that introduced fully formed organisms to Earth. Alternatively, it might indicate that life arose only once on Earth, and all subsequent evolution branched from that single origin event. The genetic evidence alone cannot distinguish between these possibilities. More intriguingly, some aspects of terrestrial biochemistry seem oddly arbitrary from a purely chemical perspective. Life's preference for left-handed amino acids and right-handed sugars appears random, yet it is universal among Earth organisms. The genetic code itself uses only 20 amino acids out of hundreds that are chemically available, and this restriction seems to have no obvious optimization benefits. These features might reflect the biochemical quirks of organisms that evolved elsewhere and were then transplanted to Earth intact, but they might also represent historical accidents that became locked in during Earth's early biological evolution. The genetic evidence can be interpreted to support multiple scenarios, including both terrestrial origin and various forms of panspermia. Isotopic analysis has provided some of the most intriguing evidence relevant to panspermia hypotheses. Different elements exist in multiple forms called isotopes, which contain the same number of protons but different numbers of neutrons. Biological processes often preferentially use certain isotopes over others, creating distinctive chemical signatures that can persist in rocks for billions of years. Carbon isotope ratios in ancient rocks show patterns that might indicate biological activity as early as 4.1 billion years ago. These signatures appear in some of the oldest rocks on Earth, suggesting that life was already influencing global chemistry at very early times. However, non-biological processes can also create similar isotope patterns, making these signatures difficult to interpret definitively. More compelling are recent discoveries of isotope patterns that don't match typical Earth signatures. Some ancient organic compounds show isotope ratios that are more consistent with formation in space than on Earth's surface. These findings might indicate that early Earth received significant quantities of organic material from extraterrestrial sources, providing raw materials for life's origin, or perhaps even delivering life itself. Sulfur isotopes provide another line of evidence. Before approximately 2.4 billion years ago, Earth's atmosphere contained virtually no oxygen, creating distinctive sulfur isotope patterns in rocks from that era. The transition to oxygen-rich conditions marks one of the most dramatic changes in Earth's history, caused by the evolution of photosynthetic organisms that began producing oxygen as a waste product. But some researchers have noted that this transition seems surprisingly abrupt in the geological record. Instead of a gradual increase in atmospheric oxygen, the change appears to occur over a relatively short time period. This pattern might indicate that oxygen-producing organisms arrived suddenly, perhaps through panspermia processes, rather than evolving slowly from pre-existing Earth life. The meteorite evidence provides perhaps the most direct information relevant to panspermia hypotheses. Over 100 confirmed Martian meteorites now reside in laboratories worldwide, each one a direct sample of material from another planet. Analysis of these rocks has revealed organic compounds, mineral assemblages formed in the presence of water, and structures that some researchers interpret as possible fossils. The organic compounds found in Martian meteorites include amino acids, the building blocks of proteins, and complex carbon molecules that are difficult to explain through purely chemical processes. Some of these compounds show isotope ratios that differ from typical terrestrial organics, suggesting they formed under different conditions than those found on Earth. But the interpretation of these organic signatures remains contentious. Critics argue that the compounds might result from contamination with terrestrial organic material after the meteorites landed on Earth. Others suggest that non-biological chemical processes on Mars could have produced the observed molecules without requiring living organisms. The microscopic structures found in some Martian meteorites present similar interpretive challenges. Electron microscope images show features that resemble bacterial cells, complete with what appear to be cell walls and internal structures. But these features are much smaller than typical Earth bacteria, leading many scientists to dismiss them as mineral artifacts rather than biological remains. Recent advances in analytical techniques have allowed researchers to examine these structures in greater detail. Some show chemical compositions and organizational patterns that are more consistent with biological origin than with purely geological processes. Others contain mineral phases that typically form only through biological activity on Earth. Yet even the most suggestive features remain open to alternative explanations. The boundary between biology and geology becomes blurred at microscopic scales, where complex mineral processes can create structures that mimic biological forms. Without clear criteria for distinguishing between biological and non-biological origins, the meteorite evidence remains ambiguous. Terrestrial extremophile research has provided crucial information about the limits of life and the conditions under which organisms might survive space travel. The discovery of bacteria living in environments once thought completely sterile has expanded our understanding of where life can exist and what it can endure. These findings have direct relevance to panspermia because they demonstrate that organisms can indeed survive many of the conditions expected during interplanetary or interstellar travel. Radiation-resistant bacteria can withstand doses that would sterilize most environments. Thermophile organisms thrive at temperatures that would kill most known life forms. Psychrophile bacteria remain active at temperatures well below freezing. But survival experiments also reveal the limits of biological endurance. Even the most resistant organisms accumulate damage over time when exposed to space conditions. Survival rates decrease with exposure duration, and very few organisms can remain viable for the millions of years required for interstellar travel. The experimental evidence suggests that panspermia is possible but challenging, requiring specific conditions and protection mechanisms. The astronomical evidence provides the broadest context for evaluating panspermia hypotheses. Observations of other stellar systems have revealed that organic compounds are ubiquitous throughout the universe. Radio telescopes have detected over 200 different molecules in interstellar space, including complex organic compounds that are essential for life. Infrared observations of star-forming regions show that organic chemistry begins operating even before planets form. Young stellar systems are surrounded by disks of gas and dust that contain amino acids, nucleotide bases, and other biological building blocks. These observations suggest that the raw materials for life are distributed throughout the galaxy, providing potential resources for any panspermia processes that might operate. The discovery of thousands of exoplanets has revealed that planetary systems are common around other stars. Many of these systems contain planets in habitable zones where liquid water could exist on planetary surfaces. Some show atmospheric compositions that might support life as we know it. But the detection of actual life around other stars remains beyond current observational capabilities. While we can identify potentially habitable exoplanets, we cannot yet determine whether they actually harbor living organisms. Future space telescopes might detect atmospheric signatures that indicate biological activity, but such observations will require technological advances that are still years or decades away. The absence of detected extraterrestrial life presents a challenge to some panspermia scenarios. If life spreads readily throughout the galaxy through natural or directed panspermia processes, we might expect to see evidence of biological activity around nearby stars. The apparent silence of the cosmos might indicate that life is much rarer than panspermia hypotheses suggest. Alternatively, the absence of detected life might reflect the limitations of our current observational capabilities, rather than the actual rarity of biological activity. Life might be abundant throughout the galaxy, but remain invisible to our current instruments and detection methods. The question will likely remain open until we develop more powerful tools for searching for extraterrestrial organisms. The cumulative evidence paints a complex picture that neither definitively supports nor refutes panspermia hypotheses. Life appeared on Earth very early and very rapidly, possibly too quickly for conventional origin scenarios. Organic compounds are abundant throughout the universe and regularly delivered to planetary surfaces by meteorites and comets. Some organisms can survive conditions that approximate those expected during interplanetary travel. Yet direct evidence for extraterrestrial life remains elusive. The structures and compounds found in meteorites can be interpreted multiple ways. The rapid appearance of life on Earth might reflect favorable early conditions rather than external seeding. The universality of biochemical features might indicate terrestrial origin rather than cosmic distribution. This ambiguous evidence base has allowed panspermia to persist as a legitimate scientific hypothesis, while preventing it from achieving definitive acceptance. Like quantum mechanics or relativity theory, panspermia remains a framework for understanding observations rather than a proven fact about life's history. The ongoing ambiguity reflects both the inherent difficulty of studying events that occurred billions of years ago and the limitations of current analytical techniques. Future advances in analytical chemistry, space exploration, and astronomical observation might eventually provide the decisive evidence needed to resolve panspermia hypotheses one way or another. Until that evidence emerges, panspermia will remain what it has always been, a fascinating possibility that challenges our assumptions about life's place in the universe while remaining tantalizingly beyond definitive proof or disproof. The evidence we have keeps the door open to cosmic possibilities while reminding us of the enormous challenges involved in understanding life's deepest mysteries. On February 11th, 2023, astronomers at the James Webb Space Telescope announced a discovery that sent ripples through the scientific community. In the atmosphere of exoplanet K2-18b, located 124 light-years from Earth, they had detected water vapor, methane, and carbon dioxide in concentrations that suggested something extraordinary, a world that might harbor conditions suitable for life as we know it. The announcement sparked intense debate about what such atmospheric compositions might mean. Were we looking at the chemical signatures of a living world? Could biological processes on a distant planet be influencing atmospheric chemistry in ways we could detect across interstellar space? The questions were profound, but the implications went even deeper. If life existed on K2-18b, what would that mean for our understanding of biology's place in the cosmos? The significance of such discoveries extends far beyond simple scientific curiosity. Panspermia, whether proven true or false, fundamentally alters how we understand life's role in the universe and our own place within the cosmic story. It transforms biology from a planetary curiosity into a universal phenomenon, with implications that reshape fields ranging from astrobiology to philosophy to space exploration policy. If panspermia operates throughout the galaxy, then the universe becomes a fundamentally different place than we have traditionally imagined. Instead of isolated pockets of life struggling against vast, sterile voids, we inhabit a cosmos where biology represents a basic feature of cosmic evolution, as natural and inevitable as star formation or planetary accretion. Life becomes woven into the fabric of the universe itself, a thread that connects every habitable world in an unbroken web of biological activity. This vision transforms our understanding of rarity and uniqueness. For centuries, humans have viewed life as an unlikely accident, a statistical anomaly so improbable that it might exist nowhere else in the observable universe. Earth became special, not just as our home, but as possibly the only world where complex chemistry achieved self-awareness and began contemplating its own existence. Panspermia shatters this cosmic loneliness. If life spreads naturally between worlds, then biology might be as common as water or carbon compounds. Every stellar system could harbor the descendants of ancient biological networks. Every potentially habitable planet might receive regular deliveries of genetic material from cosmic neighbors. The universe transforms from a largely dead expanse punctuated by rare living islands into a vast ecosystem where life and non-life interact on scales we are only beginning to comprehend. This transformation affects how we search for extraterrestrial life. Traditional SETI projects have focused on detecting signals from technological civilizations, assuming that intelligence represents life's highest and rarest achievement. But if panspermia creates galactic biological networks, then we might expect to find evidence of life almost everywhere we look for it, with sufficient sensitivity. The atmospheric signatures detected on exoplanets take on new meaning in a panspermia context. Instead of representing independent genesis events on distant worlds, unusual atmospheric compositions might indicate participation in galaxy-wide biological processes. Planets might influence each other's atmospheric chemistry through biological exchange mechanisms operating over cosmic timescales. This perspective suggests new strategies for astrobiological research. Instead of searching for completely alien life forms, we might look for organisms that share fundamental biochemical features with terrestrial biology. Panspermia predicts that life throughout the galaxy might use similar genetic codes, similar metabolic pathways, and similar cellular structures because they all descended from common ancestral populations. Such searches have already begun to influence space exploration priorities. NASA's Perseverance rover on Mars is specifically designed to look for biosignatures that might indicate past or present biological activity. But the mission's protocols assume that any Martian life might be related to terrestrial organisms through panspermia processes, requiring careful procedures to distinguish between indigenous Martian biology and contamination from Earth. The Europa Clipper mission, scheduled to explore Jupiter's ice-covered moon, operates under similar assumptions. Europa's subsurface ocean might harbor organisms related to terrestrial extremophiles through ancient panspermia events. The mission will search for chemical signatures that might indicate biological activity while remaining alert to the possibility that any detected life might share evolutionary history with Earth organisms. Future missions to Enceladus, Titan, and other potentially habitable worlds in our solar system will grapple with similar questions. If panspermia operates within our solar system, then the discovery of life on any world raises immediate questions about its relationship to life elsewhere. Are we looking at independent genesis events or connected biological networks that span multiple worlds? The implications extend beyond our solar system to the broader universe. The Breakthrough Listen project, which searches for signals from technological civilizations, might need to expand its scope to include biological signals that don't require intelligence. If panspermia creates galaxy-wide biological networks, then advanced radio telescopes might detect the chemical signatures of these networks operating on interstellar scales. Future space telescopes with unprecedented sensitivity might observe biological processes operating between stellar systems. Spectroscopic analysis of interstellar dust clouds might reveal organic compounds that show signs of biological processing. Detection of unusual isotope ratios in cosmic ray particles might indicate biological activity in distant regions of the galaxy. These possibilities are transforming how astronomers design and operate their instruments. The James Webb Space Telescope's atmospheric analysis capabilities were developed partly to search for biosignatures on exoplanets. Future telescopes will possess even greater sensitivity to biological indicators, potentially revealing whether panspermia processes operate throughout the observable universe. But panspermia's implications extend far beyond scientific research into philosophical and ethical territories that challenge our most basic assumptions about consciousness, purpose, and responsibility. If we are products of cosmic biological processes rather than terrestrial accidents, then our relationship to the universe itself requires fundamental re-evaluation. The philosophical implications begin with questions of identity and origin. Human consciousness has always struggled with existential questions about where we came from and why we exist. Religious traditions have provided various answers involving divine creation or cosmic purpose. Scientific materialism has generally suggested that consciousness represents an accidental byproduct of random evolutionary processes on an otherwise meaningless planet. Panspermia offers a middle path that preserves scientific materialism, while suggesting that consciousness might represent a more fundamental feature of cosmic evolution. If biological networks span the galaxy, then the emergence of intelligence and self-awareness might be inevitable outcomes of universal processes rather than unlikely accidents confined to isolated worlds. This perspective suggests that consciousness serves a cosmic function beyond mere survival and reproduction. Perhaps intelligent beings represent the universe's way of understanding itself, nodes in a vast information processing network that spans space and time. Our scientific discoveries, artistic creations, and philosophical insights might contribute to a galactic conversation that has been ongoing for billions of years. Such ideas remain highly speculative, but they illustrate how panspermia might reshape fundamental questions about meaning and purpose. Instead of being cosmic accidents struggling to find significance in an indifferent universe, we might be participants in processes so vast and complex that we can barely comprehend our role within them. The ethical implications are equally profound. If life spreads naturally throughout the universe, then humanity bears responsibilities that extend far beyond our planetary boundaries. Our actions might influence biological processes operating on galactic scales. Our space exploration activities might disrupt ancient panspermia networks or contribute to their expansion. The question of planetary protection takes on new dimensions in a panspermia context. Current protocols are designed to prevent Earth organisms from contaminating pristine extraterrestrial environments. But if those environments already participate in biological exchange with Earth through natural panspermia processes, then the definition of contamination becomes much more complex. Should we treat Mars as a sterile environment that must be protected from terrestrial biology? Or should we recognize it as part of a solar system-wide biological network that naturally exchanges genetic material with Earth? The answers to these questions will influence how we explore other worlds and whether we allow human colonization of potentially habitable environments. The prospect of human expansion into the galaxy raises even more complex ethical considerations. If we develop the capability to travel between star systems, should we deliberately spread terrestrial life to suitable planets? Would such actions represent continuation of natural panspermia processes or artificial interference with cosmic biological networks? Some researchers have already begun advocating for directed panspermia projects that would use human technology to accelerate natural biological distribution processes. These proposals suggest sending specially designed microorganisms to potentially habitable exoplanets, seeding them with terrestrial biology that might eventually evolve into complex ecosystems. Critics argue that such projects represent unprecedented hubris, potentially destroying unique evolutionary experiments that might be ongoing on distant worlds. They worry that human-directed panspermia might homogenize galactic biology, reducing cosmic biological diversity to terrestrial biochemical patterns. These debates reflect deeper questions about humanity's role in cosmic evolution. Are we passive observers of universal processes, or active participants with the power and responsibility to influence biological development throughout the galaxy? Should we aim to preserve existing biological networks, or should we actively contribute to their expansion and diversification? The answers will likely influence human civilization's long-term development trajectory. A species that sees itself as part of galactic biological networks might pursue very different goals than one that views itself as isolated on a single planet. Space exploration, genetic engineering, and artificial intelligence research might all be redirected toward enhancing our participation in cosmic biological processes. Educational implications also deserve consideration. If panspermia represents a plausible explanation for life's distribution throughout the universe, then biology education should incorporate cosmic perspectives from the beginning. Students should learn to think about evolution and ecology not just as planetary processes, but as universal phenomena that operate across space and time. This expanded perspective might inspire new generations of scientists to pursue research questions that transcend traditional disciplinary boundaries. Astrobiology, cosmochemistry, and exoplanet science might become as fundamental to biological education as genetics and molecular biology are today. The economic implications of panspermia discoveries could be equally transformative. Confirmation that life spreads naturally between worlds might accelerate investment in space exploration and biotechnology research. Industries might develop around harvesting biological resources from other worlds or designing organisms for space-based applications. Pharmaceutical companies might search for novel compounds in extraterrestrial organisms, potentially discovering medical treatments that evolved under completely different environmental conditions. Agricultural researchers might develop crops that incorporate genetic material from extremophile organisms capable of surviving in harsh space environments. The space industry itself might be revolutionized by panspermia discoveries. Instead of treating space as a sterile environment that must be carefully protected from biological contamination, we might recognize it as a natural habitat for specially adapted organisms. Space stations and interplanetary spacecraft might be designed as biological ecosystems rather than sterile mechanical environments. These possibilities remain speculative, but they illustrate how panspermia might influence technological development and economic activity throughout human civilization. The discovery that life operates as a cosmic phenomenon could trigger innovations and investments that reshape entire industries. As we prepare to examine the final implications of panspermia for our cosmic identity, we must recognize that the hypothesis represents far more than a scientific curiosity about life's origins. It embodies a fundamental shift in perspective, that could transform how human civilization understands its place in the universe and its responsibilities to the cosmic community of which it might be part. Whether panspermia proves true or false, the mere possibility has already begun to influence how we search for life elsewhere, how we explore other worlds, and how we think about our own significance in the vast cosmos. The questions it raises about biological networks, cosmic evolution, and galactic ecosystems will continue to challenge and inspire researchers for generations to come. The Voyager 1 spacecraft is dying. Launched in 1977, this remarkable machine has traveled farther from Earth than any human-made object in history. As it drifts through interstellar space, more than 24 billion kilometers from home, its power supply slowly weakens and its instruments gradually shut down one by one. Soon, perhaps within the next decade, Voyager 1 will fall silent forever, joining the vast community of cosmic debris that fills the space between stars. But Voyager carries something that will persist long after its electronics fail and its structure deteriorates. Mounted on its side is a golden record containing sounds and images from Earth. Music from Bach and Beethoven, greetings in 55 human languages, photographs of people and places and creatures from our world. If any alien civilization ever encounters this wandering messenger, they will discover not just evidence that intelligence once existed around a distant yellow star, but a sample of the cultural and biological diversity that flourished on a small blue planet. The golden record represents humanity's first deliberate attempt at interstellar panspermia, though we carry information rather than living organisms. We have become a species capable of spreading our influence beyond our home world, of seeding the galaxy with traces of terrestrial civilization. Whether we recognize it or not, we have already begun the process of cosmic biological expansion that panspermia theories predict for sufficiently advanced civilizations. This transformation from cosmic recipients to cosmic contributors marks a profound moment in the history of life on Earth and perhaps in the universe itself. For 4 billion years, our planet received deliveries of organic compounds and possibly living organisms from space, participating passively in whatever panspermia processes might operate throughout the galaxy. Now, for the first time in terrestrial history, Earth organisms possess the capability to reverse that flow, to send biological material and information outward to other worlds. The implications are staggering. We stand at a threshold that may represent one of the most significant transitions in the history of life itself. The moment when biology becomes truly cosmic, when living systems transcend the boundaries of individual planets and begin to operate on galactic scales. Whether life began here or arrived from elsewhere, we are becoming the agents of its further distribution throughout the universe. This realization forces us to confront fundamental questions about our cosmic identity and responsibilities. Are we the biological heirs of ancient panspermia processes, carrying forward a tradition of life distribution that has operated throughout galactic history? Or are we unique pioneers, the first species in our region of space to achieve the capability for deliberate interstellar biological expansion? The answer may depend partly on discoveries we have not yet made. Future missions to Mars, Europa, Enceladus, and other worlds in our solar system may reveal whether life exists beyond Earth and whether it shares evolutionary relationships with terrestrial organisms. The Mars Sample Return mission, scheduled for the late 2020s, will bring Martian rocks and soil back to Earth for analysis with instruments far more sophisticated than anything we can send to other planets. If those samples contain clear evidence of past or present Martian life, the discovery will transform our understanding of biology's cosmic distribution. Confirmation that life arose independently on Mars would suggest that biological genesis represents a common outcome of planetary evolution. Discovery that Martian organisms are related to terrestrial life would provide the strongest evidence yet for panspermia processes operating within our solar system. The James Webb Space Telescope and its successors will extend this search to planets orbiting other stars. These instruments can analyze the atmospheric compositions of distant worlds, searching for chemical signatures that might indicate biological activity. Oxygen, methane, water vapor, and other compounds can reveal whether planets harbor the kinds of chemistry associated with living systems. But the detection of such biosignatures will raise as many questions as it answers. If we discover atmospheric evidence for life on nearby exoplanets, how can we determine whether those organisms arose independently or represent part of galactic biological networks connected through panspermia processes? The answer may require observations and analyses that push our technological capabilities to their absolute limits. Meanwhile, advances in genetic sequencing and molecular biology continue to reveal new details about terrestrial life's evolutionary history. Every year brings discoveries of new extremophile organisms that expand our understanding of life's limits and capabilities. Some of these organisms show biochemical features that are difficult to explain through conventional evolutionary processes, raising intriguing questions about whether they might represent traces of ancient panspermia events. The recent discovery of organisms that use arsenic in place of phosphorus in their DNA, though later disputed, illustrated how terrestrial biology continues to surprise us with unexpected biochemical innovations. Each new discovery forces us to reconsider our assumptions about what life requires and where it might be able to survive. These ongoing discoveries occur against the backdrop of humanity's expanding capabilities for space exploration and genetic engineering. Private companies now launch regular cargo missions to the International Space Station. Multiple nations are planning crewed missions to Mars within the next 2 decades. Robotic probes are being designed to explore the subsurface oceans of Jupiter's and Saturn's moons. At the same time, synthetic biology has reached the point where scientists can design and create entirely artificial organisms tailored for specific purposes. Researchers have constructed bacterial genomes from scratch, engineered microorganisms that can survive in extreme environments, and developed biological systems that can perform functions impossible for natural organisms. These capabilities suggest that humanity might soon possess the tools necessary for deliberate panspermia projects. We could potentially design organisms specifically adapted for interplanetary or interstellar travel, engineer biological systems capable of establishing themselves on alien worlds, and develop technologies for delivering such organisms to distant planets. But should we? The question goes to the heart of what it means to be a cosmic civilization. If panspermia represents a natural process that has been operating throughout galactic history, then human participation might simply represent the continuation of ancient biological traditions. We would be following in the footsteps of countless previous civilizations that spread life throughout their regions of space. Alternatively, if life's distribution throughout the galaxy has been primarily accidental, then deliberate human seeding efforts would represent something unprecedented. The first time that intelligence has guided biological expansion on cosmic scales. We would be initiating entirely new phases of galactic evolution, transforming the universe from a collection of isolated biological experiments into a unified living system. The ethical implications are profound either way. Seeding sterile worlds with terrestrial biology might be seen as a generous gift, bringing the possibilities of evolution and consciousness to otherwise dead environments. But seeding worlds that already harbor indigenous life could represent biological imperialism on a cosmic scale, potentially destroying unique evolutionary experiments that have been ongoing for millions or billions of years. These considerations will likely become more pressing as our space exploration capabilities advance. Within the next century, humans may establish permanent colonies on Mars, the Moon, and perhaps other worlds in our solar system. These colonies will inevitably introduce terrestrial organisms to alien environments, whether deliberately through agriculture and ecosystem engineering, or accidentally through contamination. The biological consequences of human space colonization will unfold over timescales that dwarf human civilization's current age. Organisms introduced to Mars or other worlds might evolve in directions impossible on Earth, developing new biochemical capabilities and ecological relationships. Given millions of years, they might diversify into entirely new forms of life that bear little resemblance to their terrestrial ancestors. Such evolutionary experiments could provide invaluable insights into life's possibilities and limitations. Comparing biological development on different worlds might reveal universal principles of evolution and ecology that are impossible to discern from studying terrestrial life alone. We might discover that life follows predictable patterns regardless of planetary environment, or we might find that each world nurtures unique biological innovations impossible elsewhere. But these potential benefits must be weighed against the possibility that our expansion might interfere with ongoing biological processes we don't yet understand. If panspermia networks already connect solar system worlds through natural exchange processes, then human activities might disrupt delicate biological relationships that have been developing for billions of years. The precautionary principle suggests that we should proceed carefully, gathering as much information as possible about existing biological systems before deliberately introducing new organisms to alien environments. But the exploratory imperative argues that we can't understand these systems without studying them directly, which requires accepting some level of risk and potential disruption. The balance between caution and exploration will likely vary depending on what we discover about life's current distribution in our solar system and beyond. If we find that Mars and other worlds are truly sterile, then concerns about biological contamination become less pressing. But if we discover thriving ecosystems in subsurface oceans or hidden biological networks connecting multiple worlds, then our expansion strategies will require much more careful consideration. Future space exploration missions are being designed with these possibilities in mind. Planetary protection protocols are becoming more sophisticated, incorporating new understanding of how organisms might survive in space environments and establish themselves on alien worlds. But these protocols assume that we understand the biological systems we're trying to protect, an assumption that may prove over-optimistic. The search for extraterrestrial intelligence adds another layer of complexity to these considerations. If technological civilizations are common throughout the galaxy, then panspermia processes might be heavily influenced by intelligent activities rather than purely natural mechanisms. The universe might be filled with the biological projects of countless advanced species, each pursuing their own vision of how life should develop and spread. In such a cosmos, human expansion into space would represent entry into a community of cosmic civilizations that has been shaping galactic biological evolution for millions or billions of years. We would need to learn the rules and customs of this community, understanding our place within existing biological networks rather than simply imposing our own vision on sterile worlds. The detection of extraterrestrial intelligence would transform panspermia from a scientific hypothesis into a practical policy issue. We would need to develop diplomatic protocols for biological expansion, negotiating with other civilizations about how to share galactic biological resources and coordinate seeding efforts. The universe might require biological zoning laws and cosmic environmental protection agencies. These scenarios may seem fantastical, but they follow logically from the basic panspermia premise that life spreads naturally throughout the universe. If biology operates on galactic scales, then sufficiently advanced civilizations will inevitably encounter the biological projects of their neighbors. Learning to coexist and cooperate in such environments might represent one of the most important challenges facing any technological species. As we contemplate these cosmic possibilities, we must remember that they all depend on questions we cannot yet answer definitively. We don't know whether life exists elsewhere in our solar system. We don't know whether panspermia processes have operated throughout galactic history. We don't know whether technological civilizations are common or rare in the universe. But the mere possibility that we live in a biologically interconnected cosmos has already begun to influence how we think about our place in the universe and our responsibilities to future generations. Whether life began on Earth or arrived from elsewhere, we are becoming the conscious agents of its further development and distribution. This represents both an awesome opportunity and a profound responsibility. We possess the capability to spread life and consciousness throughout the solar system and eventually throughout the galaxy. We can seed sterile worlds with the biological innovations that evolution has produced on Earth. We can ensure that the flame of consciousness continues to burn, even if individual civilizations fail or disappear. But we also possess the capability to make terrible mistakes, to destroy unique biological experiments, to homogenize galactic life around terrestrial biochemical patterns. The same technologies that enable us to spread life throughout the cosmos could also be used to eliminate biological diversity and reduce the universe's creative potential. The choices we make in the coming decades and centuries will echo throughout galactic history. If we choose wisely, we might become gardeners of the cosmos, nurturing biological development on countless worlds and fostering the evolution of new forms of life and consciousness. If we choose poorly, we might become cosmic vandals, destroying irreplaceable biological heritage in our eagerness to spread our own biological patterns. The panspermia hypothesis, whether ultimately proven true or false, has already taught us something profound about life's place in the universe. It has shown us that biology might operate on scales we previously couldn't imagine, connecting worlds and civilizations in networks that span space and time. It has revealed that we might be part of something far grander than we ever dared hope. A universe where life represents not an accident, but a fundamental feature of cosmic evolution. If life is everywhere, then Earth is not an exception, but a voice in a cosmic chorus. We are not alone, struggling to understand our origins in isolation, but part of a vast community of living systems that stretches from the smallest microbe to the largest galactic structure. Our discoveries about life's cosmic connections are really discoveries about ourselves, revelations of our membership in a universe that may be far more alive than we ever imagined. Whether that universe has been singing for billions of years, or whether we are among its first voices, remains to be discovered. But either way, we have the privilege and responsibility of adding our own unique harmonies to whatever cosmic symphony is already playing among the stars. The music we create may resonate throughout the galaxy for billions of years to come, inspiring biological innovations and conscious reflections on worlds we will never see around stars that have not yet been born. This is the true significance of panspermia, not just as a scientific hypothesis about life's origins, but as a vision of what life might become when it finally recognizes its cosmic nature and accepts its role as a creative force in universal evolution. We are no longer just inhabitants of Earth, but potential citizens of the cosmos, ready to take our place in whatever grand biological adventure awaits us among the infinite stars.